These effects are manifest across scales, so from changes in gene expression all the way to changes in ecosystem functioning. And this broad range of impacts of drought is also reflected in the breadth of the disciplines that have focused on studying drought tolerance. So even just in this room, we have researchers who focus on drought tolerance from a perspective of plant breeding, plant biology, physiology, evolutionary ecology and genetics, and ecosystem science. So these diverse fields have made incredible progress in recent years in understanding drought tolerance, but I feel too infrequently do these different disciplines come together to share what they've been learning. And this might have to do with, we read different journals, we go to different conferences, and so we kind of felt like there was a need to bring together a broad group of drought tolerance researchers so that we might gain some new perspectives, make some new connections, and leave with a richer understanding of drought tolerance across different disciplines. So we've got a really extraordinary group of speakers here today to share their insights with us about how they've been studying drought tolerance. And I'm really looking forward to these talks. We also have some student speakers who are going to be presenting, and there's posters, so please check those out as well. But before we get into talks, I'd like to make a few acknowledgments to the people and organizations that have made this symposium possible. So first, I'll thank the USDA NEFA fellowship program that is funding myself as well as the other co-organizers of this event, Heather Cantor and Taylor Person, and Pat Byrne, who's been our fearless leader in helping us put this symposium together. We also have support from the College of Agriculture. We have support from BSPM in partnership with Cargill and Soil and Crop Science Department as well. But most importantly, our primary support is coming from DuPont Pioneer, and they've been putting on these types of symposia across the country for some time. It's a really cool program, and we're excited to be a part of this symposia series. So to learn a little bit more about that, I'd like to introduce Kevin Hayes from DuPont Pioneer. He's the team lead of genomic resources, and he's going to tell us a little bit more about this symposium and its place within the broader DuPont Pioneer Plant Science Symposia series. So thank you.
See if I can get it to come up here. Thank you very much for coming. I'm very excited to be here at Colorado State. I have a lot of enthusiasm for this symposia series, and I want to share a few slides with you to let you know what the symposia series is about, why we're doing it from a DuPont Pioneer perspective, and what it provides to the community. Really, this is a student-run organization. Everywhere that we go, every symposia series that we put on is run by students. And the only requirements that we have is that it's free, open to the public. It's run by students. It allows us to engage. Oh, good, I can walk around. So it's fully student run. We ask the students to go and find some matching funds, but it's really, themes are driven by the students. It's a way for us to engage with students, with faculty all around the world, and it's been expanding. And so if we go back, the first one was in 2008, University of Minnesota, and Cornell were both in the first year. So as you can see now, we've had over 89 events, 38 this year alone. And so this is a series that has become wildly successful, and the more we do it, the more universities become involved in it. And it really begins to build a network of students and faculty and speakers who travel from place to place. We invite students from one institution who are going to be doing the future symposia series, perhaps to an existing one, so they can see how it's run. It gives a lot of opportunities for students to connect both with faculty that are visiting and with DuPont Pioneer, begin to have that networking to see what kind of careers might be available to them. At the University of Georgia I was at earlier this year, we actually had a great dinner conversation with a group of students, and we had someone from the USDA, myself from DuPont Pioneer, and a young professor from the University of Minnesota with a group of students. And it's like, this is it, right? Government, academic, and industry. Ask anything you want. Here's your chance to figure out what are all the career opportunities. And it was a beautiful sort of experience to allow people to engage in such a manner. So in this figure alone, you can see everything in red is new this year. Right? And so including places as far as Bangalore, Australia, Europe, Africa, South America, and of course here at Colorado State. As we build, you can see the number of events by year. The slide's actually old. We've added some to this, so I believe the number this year is 38. And you can see this sort of slow growth, and it's really beginning to take off, and it's really sort of exciting to be a part of. And we start to build a network of speakers. And so you can see here in a word cloud, speakers that have presented many times versus speakers that have just once or twice. So people like Pat Schnabel from Iowa State are making a cottage industry out of speaking at these symposia series. But you see some notable names that are actually getting bigger here. Jennifer Doudna has been very involved in the Cal Berkeley Symposia series. And having someone of that stature talk about gene editing and become involved with students and speakers in this series has been a real boon, not only to Pioneer, but to the other participants of the series. And so 10 years in, we start to see a pattern. This is the network graph based on number of series. So you can see the major universities that have been in the program for 10 years that represented higher. You see Minnesota, Wisconsin, Iowa State, Illinois, Missouri, Nebraska are big components, but also Cal Berkeley, University of Florida, Texas A&M. And we're really starting to grow in South America. So I believe there are 7 symposia in South America this year alone. I will be traveling to CIOT next week to host the series there. And we are also just in the next couple weeks or previous couple weeks at Piracicaba, Viçosa, and Brasilia. But it's actually more than that. We have a series extension into China and India. We've been both to Joburg and Pretoria in South Africa, Paris this year on quantitative statistics. But again, it's student run, and the students get to choose the topics. And so the series started as the DuPont Plant Breeding Science Symposia. So not shockingly, breeding has been the predominant factor. And going back 10 years ago, DuPont Pioneer was hurting. There weren't enough plant breeders. And so the ideology of the series started off as, how can we get people more engaged with breeding activities and bring some of that talent in? You see other things start to pop up, and these are growing. So genotype or phenotyping, precision phenotyping. Genomics has been a big one. CRISPR-Cas is growing. At some points, we've had to discourage students from holding yet another CRISPR-Cas series because everyone wants to be a part of that. So if we remove sort of the top of it, you can start to see topics that are growing. My role in this, so I am a computational biologist by trade. My role in this series has been to sort of espouse data sciences at Pioneer and make data science and informatics grow on this chart. It's sort of on my performance plan for the year. How big can I make data science on this chart by next year? And so you can see the great wealth and diverse topics that we've had. And so I think the drought here is very timely, as was introduced just a moment ago. And I think it's a great sort of topic to move forward. And how can we engage drought research with all of these other topics? So I don't really have much to say on this slide. This is just, again, I really want to reiterate how much this is student-driven, how much impressed I've been with the students here and the students in other universities. Finding people with the wherewithal to, on top of their research activities, take time out to organize, to contact speakers, to host people, to walk them around campus and not get them lost. I kept trying to walk off this morning for no apparent reason. But the other portion of that is it allows, this is a graph of how students have traveled from universities to other symposia series, and begins to build a network of people, of students who are just starting out in their career, become connected all over the world with others who are organized, motivated, and involved within the plant science community. And I think the slides on the topics and the slides here really, to me, underscore a very important point. And that's about diversity. Diversity of people, diversity of talent, diversity of thought, diversity of location, and how we put all of those things together. And so when I give my data science talk as part of this series, I always show one particular slide, and I think it's very germane to the discussion today, but it's a NASA slide. And it shows the diversity in agriculture. Right? These are 6 fields from around the world, and they couldn't be more different than one another. Now I show this slide and I joke, I've actually, best we've ever had at Pioneer is someone who worked in remote sensing was able to identify 5 out of these 6 locations. I've never found anyone who could identify all 6. So this shows the diversity of all the problems that we need to solve at DuPont Pioneer, we need to solve as a plant science community. So from top left to right, the first is your mile by mile grid. It's actually upper Midwest, it's Minnesota. For this discussion today, not shockingly, image number 2, center pivot irrigation, Great Plains, that's actually far western Kansas. Image number 3, really small fields surrounded by stone walls, really old fields. This is in Germany. Lower left, this is the one that gets everyone. Collective centralized farms, so the fields are actually radiating out into the jungle from center villages. This is Bolivia. That's the one that actually gets most people. Then you have rice paddies in Thailand, and then the final image is giant 1,000-acre fields in Brazil. And so these are all of the problems that we need to solve simultaneously, and not all of them will require the same solutions. And so I think this shows to me the beauty of what it is that we're trying to do. So if you're interested in learning more about this series, we actually have a website. It's linked to from the CSU Plant Science Symposium website, or you can get back and you can see all of the different series. You can get more information about what speakers were there, what the topics were, how people were involved. A lot of them have their student committees lined up. So I'm very excited to hear the talks today on drought. I think there's a great speaker list. And so I really just want to welcome everyone and thank you for coming. And DuPont Pioneer thanks you as well. So thank you very much for your involvement.
First, just want to say thank you to the students who organized this symposium and invited me. I don't know why they invited me, but this is exciting. And when I saw the list of speakers, I was very excited to be involved and included in that group. And I'm excited to be first so I can sit back, relax, and watch everybody else's talk. And then also thanks for DuPont Pioneer for sponsoring this event as well. So I'm going to talk to you about, it's not very bright, is it? All right, I apologize. Is it possible to get just these front lights off so we can see that a little better? Well, most of the slides won't be this dark, so hopefully it won't be a big deal. That's probably good. All right, I'll come down here because I move around a lot. So I'm going to talk to you about some of the research that I've been involved in, talk about some traits that I study and think are important for understanding ecosystem function. And my subtitle here was Opportunities and Limitations for Applying Research Across kind of Agricultural and Natural or Managed Ecosystems. I'm glad I made this my subtitle because it's going to be a small part of my talk, but I was interested in this idea, especially from my PhD. I did some work with crop improvement and looking at different genotypes of crops. And as I was doing that, I was thinking about what I learned from those studies and whether they did or didn't apply to the natural grassland ecosystems that I worked in. So I think this idea of which kind of topics or traits might apply across those ecosystems, but also which ones don't apply is valuable as well. So we know like how our limits to what we should be looking at. And so I'll touch on that just a little bit. So I'll just introduce myself a little bit by talking about my research lab here. We now call the lab the grass lab. My kid was in with me at the lab one week, and he's just like, there's grass everywhere. And they drew a picture and then signed it as the grass lab and stuck it on the door. So now we are called the grass lab. So we look at ecophysiological or physiological responses of plants to changes in their environment through measuring rates of gas exchange, plant water status at different times during the day. And then we try to link that with the structure of the plant. So we look at the anatomy and how does that anatomy affect the response of plants to different environmental conditions. So these structure-function relationships is what we're interested in. And we work across a range of systems despite being called the grass lab. We've got some projects in mixed coniferous forest. We do work in sagebrush steppe in looking at juniper encroachment, mixed grass prairie, and I was saving room here for tall grass prairie because I have this great embarrassing photo of a friend I was going to put there, and then I couldn't find it. So this is blank, but we do a lot of work throughout the Great Plains and grassland ecosystems in general. So now you might be wondering, how are we the grass lab if we study all these different systems? So we've developed a scheme that we can still be called the grass lab despite what we work on. I'll come back to that slide in a second. So we realized when we were studying some ponderosa pine that really it's just a skygrass. So ponderosa pine and other conifers are now referred to as skygrasses in our lab. Deciduous or broadleaf trees are broad skygrasses, and the shrubs we study are usually woody knee grasses. So we're now taking over all different functional groups. They are all some form of grass to us in our lab. So that's how we justify being the grass lab working across these different functional groups. But that's kind of the interest that I have, and hopefully most of the students that come through the lab have, is these differences between either species or within a species, populations that vary across the landscape. So these are just populations of Butula gracilis that we've looked at some of these physiological responses in. And just trying to understand the different drought survival strategies that occur across these different growth forms and how they kind of result in the structure of the plant communities that we're working in. Okay, so what I'm going to talk about today is first I want to talk, first I'll say this. I realize as I put this talk together, I have this long build up to the punchline. So bear with me while I get through the build up. Hopefully it all makes sense and kind of building my case for a very short punchline at the end. So I'm going to talk a little bit about the classification of water use strategies and the ways we classify plants. And I think this will be hopefully familiar to all of you, but I just want to make it clear of what I'm actually looking at and how I'm defining it. I'll talk a little bit about water transport in plants because that's kind of what I focus on and what our lab is focusing on is how plants extract and move water through their vascular system. And then I'll highlight a couple key hydraulic traits of drought tolerance that I think are important and are worth spending more time looking at in both kind of natural systems and agricultural systems. And I'll try to make, again, this case of how important these traits are for us to be looking at as we move forward. So to start talking about the terminology a little bit, and this kind of highlights some of the differences between a more natural system. I'm probably going to call it more natural because all the systems I work in are managed in some way versus an agricultural system. But when we think about drought and plants kind of surviving and being successful during drought, I'm just going to use success for now as a real broad general term, and you can almost make that whatever you mean, what you want it to mean for now. But when we think about perennial species and kind of more natural systems, A lot of times we think about the survival of a population, which could be through seed banks. Those seeds surviving drought and then germinating and growing after the drought conditions have been relieved. Or on an individual basis, we have lots of species that can resprout. So they can senesce above ground tissue, resprout from that below ground tissue, and survive. Now, from an agricultural perspective, those mechanisms aren't really useful for our annual crops, and maybe for our perennial crops. They're a little bit useful, but not as much either. And so when we're thinking about kind of cropping systems, we're really thinking about what's that ability to grow during a drought. So if we're really thinking more of kind of like a growing season length. But at the same time, if we're thinking more about kind of perennial and natural ecosystems, the species that grow best during the drought tend to dominate those dry systems. So really, for both these systems, we want to know what is it about plants that allow them to grow through these dry periods of time. All right, so that's what I'm focused on, and I think that translates to lots of other more highly managed systems. And so the terms we often hear when we think about drought and plant success during drought are drought tolerance, drought resistance, or drought survival. And so I just want to go through these to make sure it's clear kind of what I'm talking about. And I know lots of the other speakers will be talking about these terms as well, so feel free to correct me or change these definitions. This will apply to my talk and possibly my talk only. I don't know. Okay, so let's talk about these different plant use strategies for just a second. But before we get into that, we have to go back just a little bit, make sure that we all remember some of this terminology and how water moves through a plant. So you've probably all seen a diagram similar to this of the soil-plant atmosphere continuum, where this water column that travels through plants is considered this continuous column from the soil up through the leaves, and then the atmosphere is the driving force for pulling that water up through the plant. And that water moves down this potential energy gradient of water, represented by the symbol psi, which is our water potential. And so you can see that our values are relatively high in the soil compared to our leaves. So our leaf water potentials are going to be more negative, drawing that water, kind of pulling that water up from the soil. So remember that leaf water potential are going to be negative numbers as we're talking through some of these results today. And as we think about leaf water potential, there's probably some generalities that you think of in how leaf water potential responds or changes in response to drought. And so some of these generalities that I want us to have in our mind today are that when we see decreasing leaf water potential in plants, we generally think of those plants as becoming more drought stressed. All right, so this is generality. We're going to talk about some exceptions to these, but anyway, that's kind of generally what we think of, right? So low water potentials mean plants are limited by water and starting to get drought stressed. We also think of plants of generally having a threshold, a leaf water potential threshold, where if leaf water potential goes below that threshold, those plants are going to dehydrate and die. All right, so there's some lower threshold that plants must stay above, or else they're going to face fatal dehydration. I'm trying to make it sound really ominous. No. And then thinking about this threshold, in order to keep water potential above that threshold, plants have to conserve water in some way, and we often think about that as being through reduced water loss as reduced stomatal conductance. And so that's kind of one mechanism that plants can use to keep water above that, or leaf water potential above that threshold. But we know there's a trade-off with conserving water, right? This is just a relationship between the rate of water loss and rate of photosynthesis. And this is just across a bunch of genotypes of sorghum. And you can see this really strong correlation. We know this in plants, that if plants are going to photosynthesize fast, they have to have their stomates wide open, and that tends to result in high rates of water loss. And if plants are going to conserve water, at some point that's going to affect their photosynthetic rate as well and reduce that growth. All right? So we have to keep this fundamental relationship in our minds as we're talking about water transport through plants. Okay, so we've got these generalities that we think of in relation to leaf water potential, but there's lots of variability around these generalities, and that's what's really interesting to me as I think about different populations and different species, and I think that's what's important in thinking about plant breeding and looking for that variability that might benefit us in increasing productivity. So now let's talk about these different strategies just briefly. There's going to be several different categories and layers of these strategies. This comes from a book by Levitt talking about how plants respond to different stresses. And he takes an approach of kind of material engineering and thinking about the stress and how plants or how materials respond to that stress. And so our first two levels of dealing with drought stress are either escapers or resistors. And so what this just means is escapers these are plants these are typically annual plants that don't even grow when drought conditions exist, okay? And when I'm talking about drought right now, I'm kind of talking about meteorological drought, where incoming precip doesn't meet the demands of evapotranspiration from the system. And so there's some plants that just survive as seed until conditions are more optimal, and then they grow quickly and complete their life cycle. So those are escapers, and for our discussion today, we're going to completely ignore escapers, and we're going to focus on resistors. These are plants that can continue growth and survive during those drought periods when water is limiting the plant growth. And so there's a couple different strategies within this resistor category that I want to talk about. The first kind of layer here is drought stress avoiders and drought stress tolerators. Now in this case, when I'm talking about avoiding drought stress or tolerating drought stress, I'm talking about the leaf water potential. Plants that are trying to keep that leaf water potential relatively high and stay hydrated, and plants that can tolerate decreases in that leaf water potential or can potentially dehydrate their leaves a bit but still maintain function. So that's kind of the contrasting part of these two strategies. And if we, I'll give you a visual here. So if we think about a system where we've got decreases in soil moisture over the course of the growing season, we might expect avoiders and tolerators to look something like this, where our avoiders are, again, trying to keep a relatively high leaf water potential. They want to keep their tissues in kind of optimal hydrated conditions, whereas our tolerators don't really care. The leaf water potential and the hydration status can decrease, and they can still maintain function during those periods of times. And so I want to contrast these two strategies a bit. And then within drought stress avoiders, there's two kind of mechanisms plants can utilize to keep that well-hydrated status or keep that leaf water potential high. And we call them either water spenders or water savers. There's lots of mechanisms within here. I don't want to talk about those too much. But we generally think of water spenders as having really deep roots that might access some more permanent supply of water, or really wide, extensive root systems that can just really thoroughly explore the soil and forage for water. And those root systems tend to help them keep that hydration status pretty high in the leaves despite there being pretty dry conditions. Water savers, on the other hand, are plants that are going to reduce water loss in some way to keep leaf water potential high. So they're plants with maybe really high water use efficiency or strong stomatal control. Or if you think about some species that roll their leaves or fold their leaves to help reduce water loss. Those are some of the mechanisms that might fall under water savers. So those are some of the different strategies. I'm really going to focus on some drought stress tolerators today, or mechanisms that provide stress tolerance to plants. But the one thing I want to point out is, I present these as really nice, discrete, different strategies, but that's not the case, right? Nature is never that clean and simple, and most plants, have some combination of these different strategies across different time scales. So there's some potential for combining different strategies within a single organism. Okay, so we're talking about drought stress tolerators now. And so what makes a plant a good drought stress tolerator? And I think this is important to understand if we want to either understand drought tolerance in a natural ecosystem, or if we want to breed plants for drought tolerance, we have to know the mechanism, right? So we can look for that. And so I'm going to start by thinking about the plant hydraulic system and how water moves through the plant. And then we'll talk about how that's sensitive to failure under different conditions. So this is just a really simple linear equation describing transpiration from a plant. And so I'm going to kind of go under this assumption that plants that can transpire at fast rates can probably photosynthesize at fast rates, right? So water use, again, linked with plant growth. And the rate of transpiration can be modeled in this simplistic form by the K, which is the hydraulic conductance of the plant tissue, times the water potential gradient between the soil and the leaf. All right, so to have high transpiration rates for a given water potential gradient, you need plants that have high hydraulic conductance. Or if we think about it in an individual plant, they need to keep hydraulic conductance as high as possible to keep the gas exchange rates high as well, regardless of what our water potential gradient is. And I'm going to present data on leaf hydraulic conductance, but hydraulic conductance is presented on stem basis, root basis, so you can measure on different tissues and kind of present it with different units. And so I'll skip that, I already said it. So just to let me go back to our equation here real quick. So thinking about a situation where K might decline in a plant, because that's what I'm going to talk about today, there's two potential responses that are going to occur in that plant. So if K declines, the plant could maintain transpiration at the same rate despite a decline in hydraulic conductance. But what that means is that as hydraulic conductance goes down, that leaf water potential is also going to decline. And we know plants are trying to keep that leaf water potential above some threshold, so this might not always be a really good strategy, depending on what plant type is and what your tissues are like. Alternatively, a plant could maintain the leaf water potential relatively constant, but that would come at the cost of reduced gas exchange rates, or in this case, reduced E. And again, we're assuming the strong linkage between photosynthesis and transpiration, so this would also suggest that we'd have a decline in photosynthetic rates if we have a reduced hydraulic conductance. So we can start to see that this transport pathway, this hydraulic conductance, is kind of fundamental to understanding how plants are going to respond under drought conditions and whether they're going to maintain high gas exchange rates or have reduced leaf water potential. So leaf hydraulic conductance is, just to give you some units for you quantitative folks out there, but it's the amount of water that can move through a tissue relative to the amount of tissue the water has to be supplied to, relative to the pressure gradient. So we have millimoles of water, or this is sometimes presented on milligrams of water, per meter squared of leaf tissue per second per megapascal. All right, so this is kind of a way of describing the efficiency which water can move through a plant tissue. This is one method that we use for measuring leaf hydraulic conductance. You can see we've removed a leaf from a branch. We've connected it to a reservoir of water that's on a balance you can kind of see in the background. And we can measure the rate of water use of that leaf tissue, then measure the water potential gradient and calculate leaf hydraulic conductance. And then we do this as plants dehydrate to see how plants lose and how fast they lose that ability to transport water through their tissues. All right, so let's just talk briefly about what is the exact pathway that water is moving through, and then we'll talk about how these systems might be sensitive to failure during dry conditions. And so in the root system, I'm going to talk about the pathway, but also what controls the rate of hydraulic conductance. So we know water is moving from outside the root in towards the vascular system, and through that process it passes both apoplastic and symplastic pathways. We likely have membrane permeability and aquaporins regulating how fast water can move into that vascular system. But at some point then it enters the vascular system, and it's really the size and number of vessel elements. I'm going to talk about angiosperms today, so we'll just talk about vessel elements. So the size and number of those is what controls how fast water can move through that tissue. Talking about the stems, these are a couple scanning electron micrographs of vascular tissue, so that there's an individual vessel element there. And so the rate that water can move through the stems is really controlled by the size and number of these cells. And then once we get to the leaves, I have a cross-section of a grass leaf. Here's a vascular bundle. Here's some metaxylem where water is coming up through. So the size and number of vessel elements in the leaf is also important. But then there's this pathway once it leaves the vascular bundle. I drew a squiggly line because we don't really know the pathway at this point. But there's some pathway through the mesophyll before that water exits through the stomate. So we have both a xylary pathway and an extraxylary pathway in leaves that we have to consider. Okay, so now we hopefully are all on the same page with these different water use strategies. We remember how important leaf water potential is and how we can use that to describe these different water use strategies in plants. So now I'm going to talk about some traits related to that ability to keep water moving through the plant that I think are important. So there's several ways that, there are several mechanisms that can lead to reduced hydraulic conductance through different tissues. The first two I'm going to talk about are solely related to the vessels and the vascular system and how water is transported through those cells. Remember that our water column in the plant is under tension. And so in this diagram here, each kind of rectangle represents one of these vessel elements. And we've got water under tension in that vessel elements, and the little gaps here in the gray lines represent the bordered pits between neighboring vessel elements. What happens when that tension gets really large, the force inward can either, the mechanism of this is a little bit undetermined right now, but essentially air gets introduced into that vessel element. Because that water column is under tension, that water quickly expands and then fills that whole vessel element. And it can actually then go between different vessel elements, filling some of the vessels with air and blocking the transport of water. All right, so when we get these embolisms formed in tissues, that reduces hydraulic conductance. So that's one mechanism that could reduce hydraulic conductance. This is a couple images from Sean Gleason, who works at the USDA ARS here in town. We're now kind of able to visualize this embolism formation on plant tissue. These are stems of corn. And so this is corn that was grown under well-watered conditions. This is corn that was droughted. And I'm going to focus on these kind of square areas. In this image under the well-watered, you can see this fairly consistent white tissue here. These images were taken with micro-CT imaging, and so they're looking for the changes in density of the tissue. And so all the cells in this area are water-filled cells. And then this plant under drought, you can see these little black areas showing up. These are vessels that are now air-filled. All right, so we can visualize the change from all of our vascular tissue being filled to having some of those vessels filled with air and embolized. And so if we zoom in, we can actually start to see the structure. These are metaxylem and protoxylem elements within those vascular bundles. So we have some way of actually observing this phenomenon happening. Another way that transport through plant tissue can be blocked is the collapse of these vessel elements. So this is from a recent paper showing, so these are vessel elements here in this top left image, and if you can see them, they kind of look like they've been squashed. Those vessel elements collapsed under the tension that was in that water column. And so then that's going to reduce the flow rate through that vessel, and it's another way that hydraulic conductance can be reduced. Again, we have a way of kind of visualizing this process in plants. Now, remember, the pathway of water movement through a leaf, we talked about being partly from the vascular system and partly that extraxillary pathway. And so what reduces that conductance through the extraxillary pathway? I don't know. That's an area of active research right now, is figuring out what that pathway is and what changes to reduce that conductance through the tissue. It could be the dimensions of that water pathway changes as cells dehydrate and change shape. It's probably partly controlled by aquaporin regulation, but we don't exactly know what's going on in that extraxillary tissue. But regardless of the mechanism, we can quantify this loss of hydraulic conductance through what we call a vulnerability curve. So on the x-axis, we have leaf water potential, so from really well hydrated to dehydrated conditions, and we have our leaf hydraulic conductance. And so we typically see some sort of, it's not always nonlinear, but we see this reduction in hydraulic conductance as these leaves dry out or as the plant tissue dries out. And we quantify, we use P50, or the leaf water potential, when a plant loses 50% of its hydraulic conductance, as a trait to describe this relationship. Now, I think the whole shape of this curve is important, but this P50 does a pretty good job of looking at differences and explaining differences across species. So we're going to look at this P50 a little bit. And we see species that have differences in the shapes of these curves and where they cross that P50 value. So this is just an example of 10 herbaceous species from the mixed grass prairie. You can see there's lots of different relationships in the shape of the curve, whether it's linear or nonlinear. The axes are not scaled the same. So we have some species that don't reach really low conductances till you get out to -3 or 4 megapascals. Some species that have hit zero by about -1.5 megapascals. The point here is that we've got a lot of variability in this trait, in this ability to keep water moving through the plant tissue. And in natural ecosystems, or more natural ecosystems, we see a pretty strong correlation between the precipitation at a site and this P50 value. So this is just for a set of 10 grass species. We see a negative correlation. So I'll have to apologize. I've got some of my axes with just megapascal and some with negative megapascals. So in this case, species on the right side of this axis, I would consider them as more drought tolerant, or they're more resistant to that hydraulic failure. And so those species with lower P50 values, those are the species that tend to occupy the dry sites. Species that have less negative P50 values, they're kind of confined or constrained to wetter sites. We see this same relationship if we look at P50 values in the leaves of tree species. So here we've got the site MAP, and we've got P50 values for a bunch of different species at those different sites. And so you can see the drier sites tend to have the most negative P50 values, and the wetter sites have species with relatively high P50 values. So this trait seems to be really important in describing where plants can live and be successful on the landscape. So we don't know all of the mechanisms of what causes the decline in K-leaf, whether it's in the vascular system or outside the vascular system, but we know that low P50 values tend to be required for plants to grow in dry environments. Plants that have high P50 values tend to be found in wetter environments. But I want to point out that there's a cost associated with this. This is a little bit of an odd way of showing it, but I told Robert, who's here today, that I really wanted to show a result from his paper that he's working on. So if we look at the two y-axes in this paper, we've got P50 values, and these are related to osmotic potential, but we can ignore that for now. And so these species lower on the axis here are those species that are more drought tolerant or more resistant to hydraulic failure. Those plants tend to have much higher leaf dry matter content, which is a way for us to quantify the investment plants make in their leaf tissue. So the point here is that if you're going to have low P50 values or be drought tolerant with regards to your hydraulic system, you're probably going to invest more in your leaf tissue or whatever tissue you're measuring. There's some cost associated with that. That's important to keep in mind. Interesting, though, there really isn't a trade-off if we think about instantaneous growth rate. So this is P50s here. Again, our more drought-tolerant species would be out here on the right, less drought-tolerant on the left. And this is maximum photosynthetic rate. We don't see a correlation there between those. And in fact, in this set of species, there's almost a slight positive relationship. I apologize, I really shouldn't have a line on there, but I wanted to highlight the lack of negative or trade-off that we might expect. So there's some ability to decouple your drought tolerance from your maximum growth rate in some species. Okay, so that's really focused on the hydraulic system. That P50 value seems to be really good at explaining where plants occur along the landscape. And so the other aspect of that, of drought tolerance, is maintaining function at low leaf water potentials. And so not only do plants have to maintain water supply under those low leaf water potentials, but the cells that are photosynthesizing and carrying on all the physiology and cool biochemistry, those also have to maintain function during those lower water potentials. And so we'll talk about a trait very briefly that kind of describes that function and that ability in plants. So now we're really focused on these cells outside of the vascular system, these mesophyll cells. Again, back to your plant physiology class, you all know about turgor, and that plant cells need to maintain turgor in order to maintain function and maintain structure. And when they lose turgor, then not only are they going to lose structure, but the function of those cells is going to decline quite rapidly. And so one way of kind of quantifying that is with pressure volume curves. These are just two examples of pressure volume curves. I forgot to go back and get the species name of this one. It's one we just did recently, and it was the lowest turgor loss point I had ever seen, so I thought I'd throw it up here. But these curves are just essentially the amount of water that's lost from plant tissue as it dries out relative to 1 over the water potential of that tissue as it dries out. And that's not really important, but what we can get from this is the turgor loss point. So these curves start out as a nonlinear curve and then switch to a linear. And so that point where it switches from nonlinear to linear is where our turgor loss point is. And you can see in these two species, we have a huge range or difference in turgor loss point. Minus 0.3 in one plant and minus 4.2 in Utah juniper. So huge range in the ability of cells to maintain function under different leaf water conditions. We historically, I think, have treated this as a fairly static trait in plants. Recent research has suggested that this is actually a very dynamic trait in some species. I've got turgor loss points here of Utah juniper and sagebrush. The dark bar represents turgor loss points I measured when plants were taken directly from the field, so they were partially dehydrated. And so we have turgor loss points of around minus 4.8. Then when I let that tissue rehydrate for about 2 hours, turgor loss point shifted by about a megapascal. So we've got a megapascal change in turgor loss point over a really short period of time. So I think turgor loss point is really important in understanding, but plants that could potentially adjust this turgor loss point on short periods of time would also be something valuable to kind of look into, but also to know before we start making decisions about how plants can tolerate drought. This trait is also really kind of effective at describing where plants are occurring on the landscape. So this data set here was a bunch of tree species and turgor loss points that were measured on those species and then grouped into different kind of ecosystems, from fairly wet ecosystems at the bottom to dry ecosystems at the top. And much like P50, you can see that plants that have high turgor loss points or relatively high turgor loss points are confined to kind of wetter regions, and this average here is for crop species. Species growing in really dry systems have a much lower turgor loss point, and there's very significant differences between many of these ecosystems. So this P50 value and turgor loss point are really good in describing where plants occur in the landscape, because if you want a plant to operate under drier conditions, they have to have these traits. You can't get away with it. These are fundamental to the plant operation. So much like P50s, we see lots of variability in this trait. It's important to understanding these species distributions, and again, low turgor loss point is required to grow in these drier environments. So I've actually done a good job of getting through my slides a little bit quickly. A couple of lessons that I think that we can take hopefully from the research on these natural ecosystems and think about how to apply them both in natural ecosystems and agricultural systems. The first thing that just want to point out is that these traits in describing where plants occur on the landscape, these are a result of millions of years of natural selection. And so if millions of years of natural selections has favored these traits, they're probably really important in understanding why plants occur in those areas. So I would argue it's a really important trait for us to be paying attention to, both from thinking about ecosystem structure and function under changing environmental conditions, but also as we're trying to improve our crop species and dealing with growing in drier environments. This might be a trait that would be really useful in understanding and improving those crops. Because for both our natural and managed or highly managed systems, we're probably going to be dealing with less water in the future, right? So regardless of how precipitation patterns might change, which are really uncertain predictions, we know that we're going to have continually increased temperatures, which is going to lead to increased evaporation from ecosystems. So just based on that, we're likely going to have reduced water in many ecosystems across the world. And so we might see shifts from species that are less drought tolerant to more drought tolerant in those systems. And so in order to make those predictions and be prepared for those types of changes, understanding these drought tolerant traits, I think, are really important in that scenario. And then again, like for highly managed systems where we're working to improve crop production under a range of different conditions, I think understanding this resistance to hydraulic failure could provide another trait to add to the suite of other important traits that folks are looking at. And I'm not suggesting this trait isn't being measured, it is, but as I look at the literature, I think it's a little less represented and could be a trait that would be beneficial in moving things forward. So just kind of along with this conclusion and driving this point home, if we think about plants that have this blue area represents kind of the safe operating conditions relative to its leaf water potential. So plants might be able to shift this curve a bit based on how it can serve water, how it roots within the soil profile. But at some point, as conditions dry out, it's going to reach this lower threshold and growth is going to stop at that point. So if we're, so then I guess alternatively, if you're a plant that has that much lower threshold, you might be able to utilize those same mechanisms of conserving water or changing your rooting profile or your root structure to more efficiently extract water and conserve it in the ecosystem. But if you've got this lower threshold, you're going to be able to operate under drier conditions that we're expecting to see in the future. So just a couple things for future directions and thinking about where this research is going and some of the, I'll highlight some of the limitations of these traits. One is we really need to identify those mechanisms of hydraulic failure in leaves. As I pointed out, there's kind of two pathways within the leaf. We don't know which one is driving the loss of hydraulic conductance. Although we worked just in the last couple of weeks at this, and so we think we have some interesting data to start looking and kind of quantifying how much of the hydraulic conductance is due to extraxillary versus xylary conductance. So I think that's coming pretty soon, that we'll be able to identify that, at least in a set of species. The other thing I think we need to work on is improving the measurements of P50 and TLP. And what I really mean here is like improving the rate of these measurements. They're pretty hard to measure. These traits take a long time. You might be able to do a few species or a few genotypes in a day. So if you're thinking about trying to quantify a whole ecosystem or lots of different genotypes, this trait right now is pretty hard to measure and would be a hard trait to implement in that type of study. However, some of the technology being used right now with imaging plant hydraulic conductance through whether they're air-filled or not air-filled vessels, I think holds some promise for speeding up these measurements. So it might become a trait that we could use for more rapid phenotyping in both natural and agricultural systems. But I think that's an area that we really need to work on for all kinds of researchers, no matter what you're interested in. And then another important trait that I didn't really talk about today, but is related to that ability to transport water through the vascular system, is Kmax. So Kmax, under a variety of conditions and water limiting conditions, it can correlate really well with maximum rates of gas exchange during the midday. So Sean, again from the USDA, has some data on maize showing that high hydraulic conductance can help plants maintain higher gas exchange during the middle of the day. It also correlates with stomatal sensitivity. So plants with high hydraulic conductance in the root system, they were able to maintain gas exchange rates even when VPD values got really high. So this is just kind of looking at stomatal sensitivity. So Kmax could be useful. Another trait that might be useful, and this might just involve measuring vessel diameters in different tissues and hold some promise for being a little bit more rapid in its ability to be used as a phenotype for understanding different plant function under different conditions. So I think that's all I have. I just wanted to acknowledge a few people. Sean Gleason again. Lots of great conversations I've had with Sean. He's provided some images. And so most of the things that I said that made sense probably came from Sean, and then everything else was me, all right? So Jesse Nippert has been involved in a lot of the research and ideas that I've shown. He's at Kansas State University. I showed data from data that was collected by grad students in my lab, and Robert Griffin Nolan, who's here as well. And then just acknowledging again that DuPont Pioneer. And at some point, it was listed that there were 3.5 students that helped organize this conference. So I'd like to thank all 3.5 of them. So, all right, that's all I've got. Thank you. I think we got time for a few questions, Patrick.
Yeah, your P50 and TLP are really interesting criteria. And you showed differences among species. How about differences within species?
Yeah,
is there good genetic variation?
There still is good genetic variation. We don't have as much of that data to see how that varies across different species, and does that relate to how big the range is of the species or not. So there is some variability there that shows promise, but I can't give you a good quantitative estimate on how much variability is there. Right now, based on kind of what I've read, it really varies across species. Some species don't show much range, some species show a lot of range. So what causes that is, I guess, the next question. So, yeah. Lisa. So for TLPs, its osmotic adjustment is really important, especially on that short-term adjustment of TLP. It seems to be driven primarily by that osmotic adjustment. If you look across a wide range of species and how much they're able to adjust osmotic concentrations, it's really small compared to the range of leaf water potentials we see. So I think it was in the same paper I showed the turgor loss point by different ecosystem types, they actually quantified osmotic adjustment. And it's on the order of 0.5 megapascal for most species when you look at well-hydrated versus droughted conditions. And we're pulling together some grass data, and it seems to be there's a greater ability to adjust osmotically. It's still less than 1 megapascal. So that it's important. So that would be over a seasonal time scale, that you could have maybe a megapascal of osmotic adjustment. So it's potentially important, but when we're thinking about difference of turgor loss point of 3 or 4 megapascals, it's not, I don't think it's the mechanism describing it across all of those species. And I don't know if that's what you're asking or not, but yeah.
Great talks.
Okay.
I was interested in your micro CT across the corn stems and the development of emboli in the vascular buns. After rehydration, do those disappear? What happens?
I'm glad you asked that because I meant to say something about that and forgot. So there is an ability to refill those embolized vessels. So plants, even though those embolisms form during the day at some point, they're able to refill those embolized vessels, and then the next day or maybe a few days later, they can continue with a higher rates of hydraulic conductance. And so exactly how that happens is a little bit uncertain still. In this study I was alluding to, a recent one that Sean and I did, we didn't see much refilling happening in leaves, but there's other examples where people have observed that refilling as well. So understanding when that happens and what species can or maybe cannot do it would be a really important thing to understand as well. So, yeah.
You mentioned something about species or plants with the low P50 and a better leaf dry matter content. Can you elaborate on that? You mean like leaf to root ratio or leaf to stem to root ratios or what?
So the leaf dry matter content is you weigh a leaf fresh, fully hydrated, then you dry it and weigh it again. And so it's just that dry weight versus fresh weight.
So it's basically somewhat its moisture content.
Yes, yeah, right. So the inverse would be that they have a lower moisture content. And so generally the way I think about that is there's a lot more cell wall material in a plant with higher LDMC, so there's generally more investment in that tissue. And LDMC correlates pretty well with like specific leaf area, but when we're talking about these hydraulic traits, leaf dry matter content has much better correlations than specific leaf area does. And why that is probably, I don't know, we could figure that out. Robert and I were talking about it earlier. Yes?
Temperature also plays a very important role in the drought stress. So how do you account for the temperature variation in terms of ecosystem level?
Yeah, so that's a really good question, is especially with turgor loss point, and the other trait we get is osmotic potential. Is that driven by precipitation or is it driven by temperature? And to be honest, those correlations are both pretty good. And we have some data within a species where it seems like it's actually driven by temperature more than precipitation. And so I think there's some things to tease apart there before we can really understand how important that is for just precipitation and how much it might be important for temperature, like freezing tolerance, really. So, I'll go Robert and then Sean.
So when water is leaving the xylem, do you think it's going through the apoplast or symplast to get, by the time it gets to the stomata, like where's the...
I think it has to go through the symplast at some point, but then the rest of the pathway could be apoplastic. There's, at least for my field, we often say like, oh, aquaporins. And I feel like that might be an important part of it, is the control of those aquaporins at some point, and that passage through the symplast at some point being controlled by the aquaporins. I don't know. I think that's definitely an area that we need to figure out what's going on there. That's going to prevent us from moving forward probably pretty soon. So, John.
That was a great question. When I think of the community of scientists that are engaged in trying to improve crop species, the performance in dry lands, by and large, I don't think the water transport traits are what they're looking at.
Mm-hmm.
They're looking at aquaporins, CO2 transporters, rubisco efficiency. How likely do you think it is to improve crop species if they're not looking at water transport.
Yeah.
Considering how fundamental it is.
Yeah, that's kind of what one of the ideas behind this figure was, that, you know, if we can, again, this curve could change, it could move to the left or the right a bit. Plants would probably want to move it a little more towards the left so they could maintain function under drier conditions, or at least maintain growth while they exploit the soil more fully. They could conserve water. That would also help them maintain growth for a longer period of time. But again, at some point, you're going to hit this lower threshold. And so I think, you know, we can tweak all those things, and those are like looking at water use efficiency or root characteristics on both ends of the water transport pathway. Those are really important in kind of increasing efficiency of growth under these different conditions. But at some point, we're going to hit this lower threshold, and I think the main way to move forward is how do we figure out how to lower that threshold so plants don't care as much what the water potential is. And I showed that one graph of P50 and photosynthesis, that there's some ability to decouple drought tolerance from your growth rate. And so we can figure out how plants do that. I think that might be the place to look for how we might lower that threshold but maintain relatively high yields under these drier conditions. So, okay, thank you very much.
He is in the Institute for Plant Genomics and Biotechnology.
Great, thank you. And thanks for the invitation to participate in the symposium. And while I'm in the Institute, my main home is in the Department of Biochemistry and Biophysics. You know how department heads, they always want you to mention that. Let me start out by acknowledging my coworkers. And I'm going to review some work we've done over many years. Most recently, this group of graduate students here, the first 4 all graduated together last spring. And that was an interesting experience to get all those people out through the system. In addition, in terms of the research scientists, Brock Weirs, who's here, has done all of the work on roots that I'm going to mention. And we've been fortunate to have funding from DOE, NSF, AFRI, USDA, and industry over the years. All right, so just to come back to that original question, what are we trying to solve? We're trying to double agricultural productivity by 2050 to meet the demands of an increasing world population that's undergoing development. And we're at the point where we're limited in terms of arable land, of course, and I believe the other main limitation is water. Now, there are other things going on that are going to make this challenge an exceptional challenge, but we're going to focus on water and land. And you'll see that we've selected sorghum to attack this problem, and I'll explain the rationale for doing that. Let me start back with crop yield plateaus. So I picked a crop, this is wheat, and you could see steady increases in yield in these good growing environments. And what happened over the last 15 years or so are these yield plateaus. That's particularly worrisome because if we need to get up here, what's going on that's constraining yield in the good environments? And there's lots of discussion about that. The other point I'll make is down here. This is Australia. Now, Australia has some of the best plant physiologists, crop breeders, scientists going, and they're working hard on wheat. But here's their yield. That has nothing to do with the science. It has everything to do with water. Okay, so what that means is notwithstanding all the expertise in this room and elsewhere, this is a really hard problem to solve. And there are going to be limits to solving it. Of course, water is essential for crop yield. Agriculture uses most of current fresh water. There's not going to be more as the fresh water in the polar ice caps melt. It's not going to be available for agriculture, of course. Irrigation systems are pretty well developed. There's lots of new technologies going on to make that more efficient.
Aquifers are being depleted. I think you know that in Colorado. We certainly do in Texas. The Ogallala Aquifer in particular, and lots of other competition. Okay, so those are sort of the ground rules of the game, I guess. If you were to, so that makes you think that we need to increase productivity without irrigating more, using annual rainfall that is very different across the U.S. So the drier regions of the country, obviously half of Texas is like that, and there's this region with pretty low rainfall, limiting rainfall, and that's where sorghum is grown mainly. And then of course regions like the Midwest, which are fantastic, until you look at high resolution and you realize even within a single producer's region, there are shallow soils or slope soils where plants suffer from water deficit routinely. Okay, so that's the water landscape. So I got thinking back, this is YB here is Yogi Berra, so it's the baseball playoff season, so I thought this was a quote that was reasonable. And this actually, I came across this, must be almost 15, 20 years ago now. And it sums up where we've been going in research in trying to overcome limitations to water. If you don't know where you're going, you're likely to end up somewhere else. Okay, so I'm going to give you a couple of examples of where when we started, we really did not understand the problem in any quantitative way. And we took some forays in directions where we ended up somewhere else. Eventually, I think we've gotten better on track, but the one place we don't want to end up is here, right? Easter Island, one of the most dramatic examples of an environmental collapse. And I don't want people to look back at the things we've done and say, wow, look at those funny statues that they built during all their research. They wasted a lot of time and effort on things. All right, so a question that we didn't ask for a long time in our research, probably 10 years in, was which plant system should we be working on? What's going to be most useful for figuring out drought tolerance research that leads to increases in crop productivity? So that's a little different. Many of you are going to talk about the ecosystem issues, and they're significant. We were narrowly focused on what can we do to increase plant productivity, crop productivity specifically, under water limiting conditions. And it led us, of course, to move to work on sorghum after spending 10 years working on Arabidopsis and soybeans and barley and a bunch of other things. So we decided on sorghum through this process. We said, well, which grass, which plant gives you the most productivity? Obviously, it's the C4 grasses because of their photosynthetic capabilities. They're used for almost everything, and there are lots of different species. This particular group of plants within the C4 grasses is particularly productive. Sorghum, miscanthus, maize, cane, switchgrass, napier grass, pearl millet, for example. And of these, there are two, sorghum and pearl millet, that are particularly drought tolerant. And I was glad that the previous speaker went through all the definitions, and I think my definitions are pretty much in line, but you'll see all of us in this field, we define what we mean because the definitions are broad, they overlap, and they've changed over time. All right, so sorghum is used for grain, forage, sugar, and biomass. So it's worldwide about 40 million hectares of grain, about the same amount of forage. Not so much acreage to biomass, but that can be used for forage. It's a C4 grass. It has a very diverse germplasm, tractable genetics, it's inbreeding and diploid, good genomics, and it's a hybrid crop. So that satisfied our needs for a good genetic system, as well as a drought-resilient grass, and one that's being used in agriculture. Now, why is sorghum drought-resilient? It's because of its history, of course. So about between 50 and 60 million years ago, the common ancestor of sorghum and the grasses diverged. And sorghum found itself in Africa, or maybe it started there, whatever happened. And over that 50 million year period, the wild species got into almost every agricultural, almost all of the ecological niches, save the Saharan desert. Okay, so the wild species are all in all sorts of environments, and a lot of those are extremely dry. So long time to do selection. And secondly, I point out that the breeding bottleneck, so sorghum was domesticated about 5,000 to 10,000 years ago, mainly here in the Ethiopian region, and was moved along the trade routes, both west and east and south, and it outcrossed with the wild species. So there's not that big domestication bottleneck. There's a lot of diversity from the wild sorghums in all of our germplasm collections. And of course, there are sorghums from India and Australia as well. Okay, lots of diversity, lots of time to select. So when you begin to think about that, you realize sorghum knows how to handle dry conditions. Let's figure out what sorghum has already learned how to do and try to pyramid those traits together and make a crop that's more productive. Now, a crop is different from a wild species. The selection pressures are different, so there's things that we have to do. All right, so here's my list of the drought spectrum, from tolerance, escape, avoidance, water use efficiency, to soil, water, and nutrient extraction efficiency. I'm going to start at the top, dehydration tolerance. And this is historically where we started. And we didn't necessarily start on sorghum, but it's the same process that we've used to look at that particular trait. So, molecular biologists that start in this field, they say, well, we can learn what a plant does by imposing a water deficit, various duration and severity, do expression profiling, sort all the pathways into biochemical, all the genes that are turned on into biochemical pathways, sort things into regulons, work our way to regulatory elements, work out the networks, and we've spent a lot of time doing that. It's really interesting research. And we built in on top of it QTL mapping, eQTL mapping, to get down to the alleles that made a difference in terms of turning on these pathways in response to water deficit. Okay, we did this back in more than 20 years ago now, and of course we didn't have RNA-seq, so we did cDNA sequencing and came across lots of interesting pathways, the ones that you would expect. Things that turn on ABA biosynthesis, hormone biosynthesis, the dehydrins, the oleosins, the protective proteins, oxidative protection in the leaves in particular, biotic resistance. When the plant shuts its growth down in response to water limitation, it goes into the protection mode. We soon realized that a lot of these protease inhibitors and other inhibitors, that's a dual mechanism. They're storing nitrogen at the same time as they're protecting the vegetative growth that's already there. And then lots of compatible solutes, proline, glycine butane, etc. There was one interesting gene we came across. It was this membrane-spanning protein. Erin Bell worked on it. She found that it was differentially induced in guard cells. Turns out later on, Martin Crispiels found that was an aquaporin. So even 20 years ago, we were able to discover things like aquaporins that were turning on in response to stress. Very interesting science. But about 10 years into this, John Boyer moved from Illinois to Texas A&M, and we began to collaborate. And for those of you that know John Boyer, he said in a very quiet way to me one day, he said, you know, those are really interesting pathways that you're discovering that are turned on in response to water deficit. But are they merely sufficient, or do they actually make any difference in the field? Are you gaining any yield under water limiting conditions? And of course, when you look at those traits and you look at dehydration tolerance, sorghum's actually, among even just the grasses, it's not very dehydration tolerant in the vegetative phase. Of course, it has to have those genes to have a dehydration tolerant seed. So in fact, when I looked at more carefully, those pathways that were being induced in response to a water deficit, yes, they're important for modulating the physiology, but the dehydration tolerance part of it, while necessary, wasn't going to contribute much to productivity. All right, that was one of many lessons we've learned along the way. I'm back to this, if you think you know where you're going, you're likely to end up somewhere else. All right, so John Boyer forced us to think about which traits. First we thought about which plant system, and next we thought about which traits are important for increasing productivity. And so I'm going to now delve into a few of those that we've looked at over the years. So one of the first we looked at was drought escape. So I spent some time with Darrell Rosenau, Fred Miller, later on Bill Rooney, the crop breeders at A&M that have been breeding sorghum. And I asked them, what makes your crops more tolerant? And they said, early flowering, that'll escape the drought. So we said, okay, well, that's a tractable trait in sorghum. Sorghum's a short day plant, very elegant work by Paige Morgan and others showed that. And there are these genotypes that if you grow them in 14-hour days, they will not flower. So it's a really strong repressing system. And as soon as you move them to day lengths that are 12 hours or less, within 10 days they initiate flowering and away they go. So it's a really interesting system. In addition, the breeders had collected mutations in this pathway called MA1 to MA6 that modulated the days to flowering. And so we set about mapping QTL, isolating the genes, and I think we've got all the genes, and we're on our way to MA4. That's the last one of this set. I'm not going to take you through that because it's been published, but we've worked out this flowering time pathway. And as you know, the shoot apical meristem is going to produce leaves and stems until it's triggered to transition to flowering. And in sorghum, there are 3 genes, the CN genes, that produce FT peptides that go to induce that transition. They're regulated by 2 activators, the grass-specific EHD1 and constans. So they activate these CN genes. And they are in turn controlled by 3 repressors, PRR37, GHD7, and CDF1. So that's the machinery. And there are alleles in those genes that affect flowering time in the breeding program. What regulates this in terms of the physiology or the incoming environment is light through the phytochrome pathway, both phytochrome B and C, and clock regulation. And sorghum uses a traditional external coincidence model to judge day length versus the clock rhythm to tell it when to turn on flowering. All right, so we worked all that out. And then we went back to the question and said, okay, guys, let's see. How early would you like us to make these plants flower? Now, the breeders had selected lines that flower at about 60 to 70 days, depending on location. And after floral initiation, it takes about 30 days to get to anthesis and then another 30 some odd days to get to grain maturity. So they were sitting here at about 60 to 70 days. It takes about 14 to 21 days to get through the juvenile phase of sorghum. And during that phase, sorghum will not flower, no matter what you do to it. And it produces about 6 to 7 leaves. We could produce plants that would produce just 2 adult vegetative leaves, and then it would trigger flowering. So 9 leaves in total. That plant will flower in 35 days. That's pretty impressive. We shorten the juvenile phase a bit. But when you look at those plants, they hardly have any leaf area, and they produce these really small panicles and only a few grain. Okay, so while we could make things flower early and escape drought, they sure didn't produce many grain. So that's adaptive in some environments, but it's not very useful for crop productivity. And that's why, of course, the breeders were out here at 60 to 70 days when they get full canopy closure, and the panicles, there's enough panicle material, enough seed sites, to use photosynthesis for the next 60 days. Okay, so again, we got going down this track. You come back to this thought. If you don't know where you're going, you're likely to end up somewhere else, and sometimes you think that's not a very useful place to be. But the corollary of this is when you get there, see if there's something you've learned that you can put to use. And in fact, this turned out to be pretty useful going in the opposite direction. So understanding the flowering time pathway has allowed us to build a breeding program. This is Bill Rooney's breeding program for high biomass sorghum. And these are very productive plants, and they turn out to be quite drought resilient. And what the understanding the alleles allowed us to do was to speed up the process of building the female lines and the male lines that both flower early so you can make the hybrid seed, but the F1 will flower late. Okay, so we got some utility out of that research. All right, now let me switch and talk about drought avoidance a bit. And in this particular case, I'm going to talk about the timing of when water is utilized during production of a grain crop, and a little bit about root angles. And this relates to a trait that was identified first by Daryl Rosenau in sorghum about 1978. And he called it sorghum stay green. And visually what it looked like is after anthesis going into the grain filling phase, these plants would tend to stay green. Their leaves would stay green longer, and that was correlated with better seed yield. And people who have measured photosynthesis show that these green leaves are photosynthetically active. It's not blocking senescence, it's keeping the photosynthetic capacity of the plant available longer, allowing more seed filling. We did QTL analysis in the 90s. There are at least 12 to 15 different QTL that have been discovered by various groups. These were all published in about the 2000 era. We now know that about half of them adjust the canopy. The other half are working on root system architecture and some other traits. We constructed near isogenic lines, NILs, so we could do some physiology. Andy Borrell published 2 really nice papers describing the physiology underlying the canopy traits for 4 of the QTL. And I won't go through that because they've been published, but I'm going to show you what it looks like because I think it's pretty impressive. So if you take 3 of those QTL, stay green QTL, and integress them into this high yielding but senescent background, then grow those plants to 14 days before anthesis and stop watering them, and let them go through anthesis. This is 10 days afterwards. There's the senescent line, and this is when you add those 3 stay green QTL into that background. Makes a difference. Easy to see. Took us a while to figure out how to do this, but if you take even a single QTL, stay green 2, grow the plants to flag leaf, under well-watered conditions, they look great. You stop watering, and again, you can tell when you have stay green 2 in that plant because it stays green and it produces more. The yield advantage under these water limiting conditions, under field conditions, is about 5%. And you say, it doesn't seem like that's much, but 5% is 5%. And once you've got it in the crop, it's going to be 5% compounded. That's pretty good. Okay, what's this gene doing? What's this locus doing? Well, it's doing 2 things. Early in growth and development of sorghum, it's reducing the amount of tillers, and that's reducing leaf area. And it's also, after floral initiation, the upper leaves that are produced are smaller. And that also reduces, well, it reduces the use of carbon for leaf growth, so that carbon can be used for grain filling, and it is also reducing the canopy architecture at the top, smaller leaves, and driving photosynthesis deeper in the canopy, and that combination of things saves water. So this, it's a really simple trait. Stay green is all about saving water from the vegetative, that would have been used during the vegetative phase, and moving it so it can be used during anthesis and the grain filling phase. So it's a water resource utilization trait. It's probably got more details under the hood, but that's the main effect. Okay, that was a bit about drought avoidance. Let's talk now about water use efficiency. And I'm going to tell you about 2 traits quickly. One is VPD limited transpiration, and the other is leaf angles. So basically, as we know, what the plant's trying to do is take light energy, extract electrons from water, release O2, make reducing power and ATP, and use that to drive the carbon cycle to fix carbon into biomass, which then can be partitioned into various things, including grain. And we know one of the issues is when you open the stomate so CO2 can be moved into the leaf, the water escapes. And the previous talk was great, described exactly the issues involved in this. What's happening, so here's just water from the soil going through the stem and into the leaf and then back out of the leaf surface. And as was pointed out in the previous talk, that driving force, the difference between the water potential in the leaf and in the air, drives water out through those stomates, and as vapor pressure deficit goes up, transpirational efficiency goes down. More water is lost for every CO2 that gets into the plant. It's a well-known interaction and response. So the question is, and it's well known to negatively affect yield. So under conditions, environmental conditions where VPD is high, crop yields go down. This happens to be corn. I know Pioneer's fields would never look like this, but at any rate, that is the issue. Question is are there any genetics that you can get around this or at least ameliorate the response? So here's that response. What one would want to do is reduce the amount of water use at high VPD, which happens in the afternoons in hot, dry environments, and save that so it can be used in the mornings when VPD is lower. Now, Graham Hammer and Tom Sinclair identified sorghum genotypes that manipulate this trait, and they have 3 components to the trait response. The first is the so-called M1 slope, which is the linear response to an increase in VPD. And some sorghum plants just keep following M1. And there's variation in that M1 slope. It's interesting that in Australia, the optimal hybrids have a lower M1 slope. They use water a little slower at high VPD than hybrids that have been selected in Texas. The second part of this trait is the break point. So at some VPD, some sorghums will stop increasing their transpiration and just plateau. And others, a very few, will actually go negative. So those are the 3 things we can manipulate to see if there's an optimum here. And you can see we're learning because instead of finding the genotypes, selecting for them, doing the research, and then asking whether it'd make any difference, we decided to model this trait first to ask how much difference could it make before we put all the effort into finding the genes and deploying these in hybrids. Now we thought this trait would be most important in these forage or high biomass sorghums because we grow them full season. They go through every summer, and many summers there's not much water available. We're able to adopt the APSIM grain sorghum model and parameterize it based on the energy sorghums, and you can see that worked pretty well. And using the model and confirmed by results, under irrigated conditions in our location, we can get upwards of 50 dry tons per hectare of biomass. That's a lot of biomass. That's fully irrigated. If we don't irrigate in College Station, you get about 20. That's the effect of water limitation. So anything we can do to help these plants use their marginal water resources and move up this yield curve is going to make a big difference. The other thing about what happens when you go through a long period of water deficit for these high biomass sorghums is they'll go on, they'll stop growing, and then they'll sit there for weeks. And then suddenly the leaves start to senesce from the bottom, and eventually they're all gone, or almost all gone. The plant's still not dead. You get another rainfall, it'll put a new flush of leaves at the top. But in this particular case, this was a 6-week drought. It lost all its leaves. By the time the rain came in the fall, the plants had to rebuild their canopies. They really weren't that productive. So we'd like to avoid this as well. All right, so took these 3 parameters, the M1 slope, M2 slope, and the VPD breakpoint. We determined that the M1 slope that the current genotypes has in Texas, it's optimal for high productivity. That makes sense. Stomates are open, they're driving photosynthesis at the maximum rates. So we didn't change that. And you can change the VPD breakpoint or the M2 slope. The VPD breakpoint, if you think about it, if you moved it down, 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 you'd end up doing less and less photosynthesis during the day. So it'd be only a couple hours in the morning and then nothing. Well, that's not very productive. So if you go below 2, you really get into losses of biomass. So we needed these VPD breakpoints to be 2 and above. There are a few sorghum genotypes that are below that. They're down at 1.6. Okay? But we thought for productivity purposes we need to be above 2, and we have several that are in that 2 to 2.5 range. The other thing you can do is change that M2 slope. So after the VPD breakpoint, do you just plateau or do you go negative? And it turns out if you can find a genotype that will go negative, that is shut down transpiration, either through shutting down its hydraulic system or stomates, that gives you a pretty broad range where the plants are productive. They'll save water. They become about 5% more efficient in terms of transpiration efficiency, biomass per unit water used. And importantly, they avoid doing this. So what happens is if you add in that negative M2 slope at the optimal VPD breakpoint, they won't lose their canopies. So for an energy sorghum, this is pretty important. So now that we've done the modeling, we're going back to find the QTL and put them in the hybrid program. Okay, briefly, let me do 2 more traits, leaf angle, and then I'll do water extraction efficiency. So leaf angle is also going to aim at the water use efficiency trait. It's been well known that photosynthesis, even C4 photosynthesis, is saturated in midsummer. There's way too much light at the top of the canopy, so it's inefficient. And breeders over the years have changed leaf angles to make them steeper, more vertical. I mean, look at any modern corn. It has a really steep angle in terms of its leaves. The other thing that does, so that moves photosynthesis deeper in the canopy. There's lower VPD in the canopy. It's cooler, and it saves some water. Okay, so again, we modeled this first before we went into looking for QTL. And Sandra Truong that did this work was getting more sophisticated. She could put plants in fields and simulate the results. A large angled leaf that is a sort of flat horizontal leaves, you can see how often the leaves are saturated in this energy sorghum field. Not in the middle where the whorl is and the leaves are vertical. This is an energy sorghum, vegetative grown. And if you just shift the leaf angle, you can eliminate most of that saturation. All right, so then we went in, found QTL in our bioenergy populations, devised 2 near isogenic lines, one that had steeper angles, one that have more horizontal leaves, and grew them out, and sure enough, the small angled leaf populations, plants, had about 10% more biomass. We haven't yet measured their water use efficiency. This was already good enough to move into the crop breeding pipeline, so as soon as we generate hybrids with different angles, we'll go back to the water use efficiency measurements. All right, so notice what's changed. We stopped guessing or asking people and then launching into a research area. Now we define a trait, we model the trait in our particular crop and environment and management practice. Once we've decided that it works, we find the QTL, and then we move them into the breeding program. At least for us, that's a lot more efficient. All right, finally, we're also working on root systems and how well do they extract both nitrogen, other nutrients, and water from the soil. I'm working on this a while. I don't have too much to show other than we know how to dig plants' roots out of the field. We can image them. There's tremendous variation, lots of different structures. We can map QTL. There's a huge variation in root traits in sorghum. More recently, we've gone to, in addition to field studies, doing these rhizotron lysimeters with the field soil, the natural microbes that are in there. That's important for normal growth and development of the root systems. And we can impose different conditions that really change the architecture of the root system. So you can see here, under this condition, the roots stay mainly at the top of the profile, and in other conditions, they proliferate through the entire soil profile. We think there's utility here because sorghum in general doesn't get its roots deep enough to get all the water out of the soil profile. And there's lots of other things going on with root systems that I think are important. So we're working on that. All right, so let me come back to this image. You notice the Maui here. They're sitting at the edge of this island, Easter Island. They're looking out to sea. That often puzzled me. So what? The Polynesians that went to Easter Island, nobody else came for, 800 years. They weren't worried about people attacking the island. Why are these guys looking out to sea? Maybe they're longing to get off the island. I'm not sure what. But it made me also think, maybe we haven't thought through this problem completely enough. And it made me wonder about this question. Which crop, and in sorghum that is grain, forage, high biomass, which crop is the most important for productivity under water limiting conditions? So of all the crops we could grow, just of the sorghum types, which one would be the best to grow? And I think, all things being equal, it's this high biomass crop. It's the most drought resilient crop that we grow of the sorghum types. It doesn't go through a flowering phase, which is the most sensitive phase of growth. it will sit there and take it. It recycles its nitrogen, its high nitrogen use efficiency, and it produces a whole lot of biomass. So now we're really thinking, this is our crop target if we want to use water most efficiently. And what we need to do is re-engineer the composition of the crop so it can be used more effectively. So in our other part of our program, we're working on the engineering of composition. And I'll stop there and just remind you, pick a good genetic model. It took us 10 years to find the one we wanted. One that has both crop and lots of applications going on. And hopefully choose the right questions to ask long before we did. And see where you get to. Anyway, thank you. Enjoyed the invitation to the symposium. Happy to take questions. Yes?
Has anybody tried to transfer the stay green trait to other crops or plants? What are the results?
So my colleagues in Australia, Andy Borrell and David Jordan, once it was clear what the stay green trait was all about, it's actually fairly straightforward to begin to select for it in let's see, parallel germplasm and other species and to screen for it. So that's going on. To tell you the truth, of the eight or so QTL that we've started doing map-based cloning on, we've only gotten to actually 2. And those are in transgenics at present to confirm that they confer the phenotype, and then they could be moved to other grasses. But generally, I mean, we know what we're looking for in terms of physiology, so I think it could be selected. Most of the stay green phenotypes could be selected in other crops. Dan?
Stay green, I assume you have to fertilize.
Grasses require nitrogen, yes. But are you asking? You must be asking that for a reason. Come on. Do you have to give it more nitrogen?
You think about the physiology genetic background. You're senescing to move that nitrogen into the grain, right? So if you're going to keep a leaf green and still get grain, you have to have another source of that.
Okay.
That's my point.
Okay. Let me answer that. That's an interesting question. So Andy Borrell and Graham Hammer wondered about that. They wondered about the nitrogen that gets to the grain. And the stay green, more nitrogen gets to the grain than the non-stay green types, even though the senescent types are senescent. The stay green.
How do you explain that?
Okay, I'm going to explain it. It's simple, actually. It turns out
You're still moving water from the soil, and therefore you're bringing the nitrogen up.
Exactly. Nitrogen fixation and uptake, assimilation, is occurring much longer in the stay green types. So they just get a lot more nitrogen for longer, and they put it in the grain. Yeah. Yeah.
For the three-dimensional figure you showed with the BP breakpoint,
Yes.
Why is there a bump in there's like an optimum BP breakpoint, and why doesn't productivity just maximize and plateau? There's sort of a bump beforehand.
Well, yeah, there is an optimization. There is an optimal peak when you take all 3 parameters and optimize them. And the reason that occurs is that's actually driven by water use efficiency that's gained by the trait under water limiting conditions. And you get about a 6%, so it's pretty flat for most of that, but goes up when you hit that maximum. And we could talk about why that is more, but it's like, okay, yeah, that's probably enough for now. Yes?
How are sweet sorghums looking in energy crop profile?
So we used to think of sweet sorghums as unusual outliers, but when we got into the energy business, we realized when you screen all the germplasm, most sorghums can accumulate sucrose in the stem. Most do, except those that produce aranciment in the stem. And we've tracked down the main genes that cause the formation of arancima. So now nearly any sorghum can be a sweet sorghum. You just stop letting them hollow out their stems. So the question is, do you want sugar in the stem when you harvest a bioenergy type sorghum? And right now the answer is yes, because that's very easy to ferment, but it has some logistics constraints. So the sugar will be turned over by microbes, so the feedstock itself is not very stable, And we've also reached maximum limits of the sugar accumulation. Sucrose is about half molar in those stems, just like sugar cane. And that's actually creating a feedback loop and shutting down photosynthesis. So we're now trying to convert that sucrose into some other carbon polymer to relieve that feedback loop. Got 3 questions here. Go ahead.
Okay, so I really appreciated the sort of introduction with it, explaining the sort of natural history of sorghum and its origins sort of historically. And I'm trying to think about sort of that, and along with the observation that there's all this genetic diversity in sorghum,
right,
and they're sort of found from all these different places. And you also identified some really interesting drought tolerance traits. And I'm curious, do we have any sense for the diversity of these traits in sorghum and its relationship to the types of places where it's found? And can we infer the sort of adaptive value of these traits with regard to local?
Great question. No doubt about it. The stay green traits, they mainly come from sorghums that originate just below in sub-Saharan Africa. There's strong selection pressure there, and that's mostly where they come from. The root system variation comes from different soil types. So the deep root systems, so there's all that. The leaf angles, I'm not sure. We haven't really gotten to enough of those genes to track them back allelically. Obviously, flowering time alleles go north-south across that gradient. So in most cases, you can trace back to the original region to find out where those alleles were coming from. Yeah, great question.
You talked about leaf angle. I know in Australia, some people have also looked at root angle as a way of getting to deeper water. Is that something you've looked at and found to be useful?
So the other half of those QTL that contribute to stay green, some of those are root angle traits. And yes, in collaboration with David Jordan and Andy Borrell, I think it was David Jordan's main work. That's an interesting trait because that one's got different optima depending on how you're planting and how much water is there. So if you have wide spacing between the rows, you actually want an angle that will get the water between the rows. But if you go at higher densities, you want the angle steeper. It actually saves a little water early in the season, but gets water deeper late in the season. So yes, root angles. We've done a lot of QTL studies on that, so we're also working on that trait. Yeah, it's a pretty easy one. Taylor.
So you mentioned modeling, and I imagine that's been pretty successful for the area around College Station. Have you been able to apply that to other environmental conditions?
Most of these models, the AppSim model, you can plug in now that we've got it set up, you could plug in your environmental parameters and run the model, and it will give you a pretty good output. AppSim is used throughout Australia on different crops. It's used by their prime minister to predict what's going on in agriculture and make economic forecasts. It's a really good model. Now, there are other comparable models, but the reason I like AppSim for sorghum is Graham Hammer developed it starting in the 60s and has been improving it steadily since then. It's parameterized and tested for sorghum over this 30-year period. So it's really a robust model. Sorry.
I have one question. What is the contribution of this QTL that you identified for this always green one? Like in terms of how many QTLs are involved in this one, for this trait that you are showed?
The stay green trait?
Yes.
Well, there have been groups in India, Australia, and our group that have worked on this, and I believe they're up to at least 15 or 16 stay green QTL. But there are some there's some QTL with main effects. And so you can get most of the benefits, and as you pyramid them together, you can see that they're redundant in a sense, some of them. So you can get the main effects with about maybe no more than 4 of these QTL. And they've been introgressed into the Australian breeding program. David Jordan does not, he's the lead breeder in Australia. No line comes out in Australia that doesn't have stay green in it. It's that important.
If I may ask one more question. Is there any potential gene has been identified, candidate gene identified in the QTL regions?
Yes. We're still confirming those.
Okay.
It took us decades to get to them. Like, oh my God, that's the worst thing that have to map-based clone ever.
Maybe one question on the leaf angle, the broad versus high angle. There's a significant effect on plant height as well. Are those inflated or?
Oh, that's an interesting observation. Most people just say, oh, it's bigger, more biomass. We thought it would affect internode lengths, but actually what's happening is the plant probably is a feed-forward mechanism, either from photosynthate, more photosynthate, or the interception of light signaling. It's producing phytomers faster, which is an interesting. So we've got starting to look at phyllocron.
So more internodes, not longer.
More internodes, more leaves, faster. It's kind of interesting. I did not expect that. Okay, great. Thank you.
All right, thank you, everyone. I'd like to thank again our first three speakers, Kevin Hayes from DuPont Pioneer, Troy Ogletree from Colorado State, and John Mullet from Texas A&M. Thanks again. We're going to take a short coffee break. We'll reconvene at 10:35 in about 15 minutes with our three student presentations.
I've been reading for 3 years, reading for driving. So anyway, so he's got a job. We're not looking at anything really specific. So yeah, I think that's what you're looking at. Trying to figure out how all this fits into the selection. It's taking all these traits and maybe combining it into one number. I don't know what trait is actually going on there. But we know deeper roots probably, or something about maybe saving water in the morning.
room have at one point either been a graduate student or are currently embarking on the equally arduous and rewarding journey of graduate school. Having the opportunity to present our research to a room of industry professionals and colleagues makes all of the late nights in the lab, the countless field hours, conferences attended, hands shaken, and printing fees paid all worthwhile. Discourse and constructive criticism are all part of that, of course. But the three students presenting today are tremendous ambassadors for the graduate student body as a whole, as well as the rigorous applied plant science research performed here at CSU every day. We appreciate their fortitude and hard work. Our first student speaker is Nate Sint. He's a PhD student studying biochemistry in Jessica Prenny and Adam Huberger's lab. Please welcome him up. Thanks.
Okay. This is really official. Can you hear me? Hi, my name's Nate Sint, and as she mentioned, I'm a PhD student in the biochemistry department. And I come from an ag background, so most of the people in my department are studying health-related issues, and so I sort of had to weasel my way into agriculture from the biochemistry department, and luckily I found Jessica and Adam that were able to find a project for me that's I've really come to love, and I'm excited to tell you a little bit about it. So my title is Mitochondrial Inhibition Elicits a Temporary Drought-like Response in Winter Wheat. And I have Dr. Courtney John on here as well. She's on my committee, and she's the head of this particular project. So as we all know, winter wheat is a very important crop globally to feed our growing population. And it's economically important here in the High Plains region, where a large number of acres of winter wheat are grown and harvested every year. And it's important to note that within this region, the majority of these acres are rain-fed and not irrigated. And as any farmer would tell you, farming is at the mercy of the environment. And so it's hard enough on a yearly basis when you have potential impacts of plant disease, as well as impacts with severe weather. But water is the main contributor to differences in yield. So shown here is a plot of maize yield in 3 different water conditions in Nebraska and Iowa. And shown here in green is irrigated regions in Nebraska and rain-fed regions in Iowa, where the precipitation is consistent and they have high yield and low coefficient of variation in this yield. But in locations like eastern Colorado and western Nebraska, where this is a rain-fed location, the yield is much lower and the coefficient of variation in this yield is also much greater. And so farmers would benefit from mitigation strategies as well as breeding strategies to under instances where there is a high drought year potentially, you can increase the slope of this line and have a higher grain yield, but still have the potential to have a much higher grain yield if the conditions are favorable. And interestingly, chemical companies will use fungicides to actually promote this risk mitigation of plant health. So not only disease or like fungicide management, but also crop protection or plant health. And so, you know, I think a lot of times we take what these companies say with a grain of salt because they're selling a product, but I'm here to tell you today, hopefully convince you that there is some credence to this. And so back in 2014, Dr. Scott Haley, who's the wheat breeder here at CSU, performed an experiment where he sprayed some plots with this BASF fungicide Preaxor and left some of the plots unsprayed across different varieties and at 2 different locations, an irrigated location and a rain-fed location. And interestingly, this Preaxor increased yield in 4 out of the 5 varieties at the rain-fed location and 1 of the 3 varieties at the irrigated location. And he repeated this experiment in 2016, and 2 out of the 5 varieties were significantly increased for yield due to Preaxor spraying versus the 3 that were not significantly upregulated. So it's also important to note that these years, under these conditions, the disease pressure for fungus was low. So it does seem that Preaxor is having some additional effect to increase yield in these different wheat varieties. Now, these fungicides are a special class of fungicides called mitochondrial inhibitors. And Preaxor contains 2 active ingredients, the first of which is a succinate dehydrogenase inhibitor, which is complex 2. And it also is involved in the TCA cycle converting succinate to fumarate. And the other active ingredient is a Q0I inhibitor, which is a complex 3 inhibitor. And both of these inhibit the flow of electrons to plastoquinone, which then reduces the ATP output by the mitochondrion and thus kills the fungus. But we also know that plants have mitochondria, and I performed a preliminary experiment looking at mitochondrial enriched cell suspension of winter wheat leaves and measured the amount of oxygen flux in this cell suspension to look at whether or not mitochondria were inhibited by Preaxor in vitro in wheat leaves. And so shown here is oxygen concentration decrease in the cell cuvette. And when we add succinate along with other substrates to begin respiration, we see that when we reach a maximal level of respiration here and then add our mitochondrial inhibitor, which is roughly the same amount at the same concentration that they would spray on the fields, it inhibits plant leaf mitochondria by about 25%. So we know it is having an impact, at least in vitro, in plant mitochondria. And as well, there's been a lot of research that has shown that this is not just specific to wheat, but across many different species, including soybean yields, shown here, where increasing amounts of mitochondrial inhibitor fungicide increase the number of pods per plant. And at a different strategy, using some transgenic tomato RNAi lines where succinate dehydrogenase was inhibited using RNAi, these lines had a greater biomass in both total leaf biomass as well as the fruit yield. At this point, you're probably wondering what this has to do with drought. But if you remember when the stomata are closed, this is saving water flux from leaving the plant leaf, but at the same time, it's decreasing the CO2 concentration that's within the leaf. And what this does as a side effect is you have a competing reaction for Rubisco with oxygen, which is the oxygenase part of Rubisco here, and this causes a wasteful metabolic pathway called photorespiration, where the substrates then have to go through these energy wasteful pathway to recycle those products so that they can be reused. And it's important to note that photorespiration involves both reactions in the chloroplast, the mitochondria, and the cytoplasm. So it sort of is a pathway that links these 2 organelles. It's also important to note here that a lot of research in the last 5 years or so has looked at the interaction between mitochondrial mutants and their effects on photosynthesis. And there seems to be a lot of mutations here that then have a photosynthetic phenotype. So there's some crosstalk and a lot of information to be gained between the interaction and the redox pathways between these organelles. So sort of the big picture question for my PhD thesis is how do these mitochondrial inhibitors increase yield? There's a lot of phenotypic information about what these mitochondrial inhibitors do, but what's happening at the molecular level, and that's what I sought to figure out. And so we went back to Scott's field data and focused our research on two varieties that were of interest to us. One we call the responder variety, which is Brawl CL+. It will be in blue for the rest of the talk. I'll probably refer to it interchangeably as Brawl or responder. And then sort of the other end of the spectrum, we chose Ripper as our non-responder variety because in the situation where most of the varieties increased yield to pre-Axor spraying, Ripper was the only one that did not. So we'll refer to it as our non-responder, and it will be in orange. So I performed a number of different experiments. They can be categorized into two categories here. Non-destructive experiments where I used a light core to measure gas exchange and fluorescence, and some destructive measurements where I looked at root and shoot biomass, leaf area, as well as quenching samples for proteomics and metabolomics. And in the non-destructive experiments, I'll be focusing on just full water condition, plus or minus pre-Axor, and looking at days post-application. So I looked at many different time points within the first 2 weeks, as well as at 28 days and at harvest. Today, with the limited time that I have left, I'm going to focus on some data that I've collected at 1 day and 7 days after spraying, mostly focusing on proteomics with a little bit of the gas exchange data. So under the full water condition, again, this is in the greenhouse, and under the full water condition, at 1 day post-application, again, this is sprayed under the same conditions that, that BASF recommends that these mitochondrial inhibitors be sprayed. We see a striking decrease in the maximum photosynthetic rate of the sprayed versus the control plants in our responder variety Brawl. But in our non-responder 1 day after spraying, we see no significant difference between our maximum photosynthetic rate of our non-responder variety. So this supports the field data in that there is a phenotypic difference here 1 day after spraying in our responder versus our non-responder variety. So we got pretty excited about that finding. Interestingly, when we performed this experiment 7 days later, we actually see that this response is transient, where after 7 days in both varieties, there's no difference of maximum rates of photosynthesis in both Ripper and Brawl. So this suggests that when spraying with this mitochondrial inhibitor, the physiological response is transiently expressed. And so what's going on in the cell that this transient phenotype is causing downstream? And so to look at this, we did a preliminary proteomics and metabolomics experiment where we took the uppermost fully expanded leaf and quenched it with liquid nitrogen, and then extracted both metabolites and proteins for proteomics and metabolomics, saving the polar fraction for non-targeted LCMS, and the insoluble protein pellet for non-targeted proteomics. And so when we take a step back and look at the entire protein and metabolite profiles, one really interesting thing to note is that if we plot the fold change of the treated versus the control metabolites and proteins, so the proteins are on the left here and the metabolites are on the right, with Brawl on the x-axis and our non-responder Ripper on the y-axis, if mitochondrial inhibition was acting in the same way on both plant tissues, we would expect that the majority, or at least a larger proportion of the points would fall on this y equals x line, where the responses would be the same. But in fact, there's really no correlation between the two varieties in response to pre-Axor, and thus this again supports the differentiating phenotypes of our responder and our non-responder. Now, if we zoom in and look at particular proteins that were changing in response to pre-Axor, we see, first we identified 611 total proteins, 31 of which were insignificant. We see proteins fall into 4 major categories. The first is central metabolism, Second is photosynthesis. The third is gene regulation and signaling. And not surprisingly, reactive oxygen species metabolism. It's known that inhibiting mitochondrial respiration will cause a large portion of reactive oxygen species metabolism. I'll touch on that a little bit later. But the most significantly regulated protein, and this was only, it looks like it's highly regulated in both, but it was only significant in our responder variety, is ferredoxin glutamate synthase. And so if you remember back to this photorespiration pathway where it wastes energy to replenish these intermediates, it also loses nitrogen here. And ferredoxin glutamate synthase is actually localized to the chloroplast, and it is essential to this reaction of the ferredoxin GOGAT pathway, where it recycles nitrogen back to from glutamine back to glutamate, so that glutamine synthetase can refix that nitrogen that's lost in photorespiration. We also know that this protein is critical for photorespiration in plants under ambient conditions, because a study was shown in Arabidopsis that if you knock out this protein, the plants are conditionally lethal at atmospheric CO2 levels, and they're only viable under enriched CO2 or low oxygen conditions. So we know that this pathway is critical in that. Another group of proteins that was highly dysregulated between the two, the responder and the non-responder, were proteins associated with photosynthesis. And interestingly, the majority of these proteins are actually involved in one pathway of photosynthesis that's called linear electron flow. And this pathway is upregulated under drought or photorespiratory conditions, where they need to increase the ratio of ATP to NADPH. And what they actually do is they recycle the NADPH back through plastoquinone to increase the amount of ATP yield that's needed to overcome the ATP loss to photorespiration. So quickly, just to summarize, I'm running out of time here, to summarize the morphology results. Again, I looked at plants from 1 to 14 days after post-application, and I looked at the amount of water that each of the pots used. And so this is summarizing across both varieties, both Brawl and Ripper. Both varieties actually decreased the water use when sprayed with Preaxor in both the full water and in the drought treatment. So the trend is in the direction of a drought here, but it's obviously not nearly as substantial. And the same with shoot biomass as well as root biomass, which I'm not showing. But so in conclusion, a few of the conclusions that I've made so far is that there is decreased photosynthesis in the responder 1 day post-application, which seems to upregulate the response of central metabolism, photosynthesis, and photorespiration in our responder variety, but not in the non-responder variety. And then some of the morphological characteristics, it's kind of interesting, Dr. Mullet, what you mentioned previously is that we also saw decreased water use, decreased root and shoot biomass in both varieties. And it's also been shown that this, one of the phenotypic mechanisms, or not mechanisms, but results that people have previously presented on is that it seems to increase healthy area duration, or this stay green phenotype as well, when you spray with this mitochondrial inhibitor. So to kind of give you the working model of what I think is going on is, I think in both varieties, the mitochondrial is being inhibited by this mitochondrial inhibitor fungicide. And downstream of that, either the plant is sensing this decrease in ATP and NADPH output by, or NADH output by the mitochondria, and especially increasing reactive oxygen species, and this has been shown. And that's causing downstream gene expression, which is then regulating both photosynthetic pathways and nitrogen fixation pathways in our responder variety, but not in our non-responder. So I think there's potentially two pathways being sensed here. One that's causing these morphological changes that is consistent between the varieties, but this biochemical difference in our responder that then gives an advantage during the season in grain filling. So I think with that, I'll take any questions. Right, so it's applied at Feeks 5, which is a late tillering stage. It's usually late April in the springtime.
Okay. So why would you expect sort of a continuing?
Aren't really apparent, and there's a large environmental effect with when I'm measuring to find these differences, because as the plants are growing, they're becoming more variable, but I think the effects that I'm looking at are at the molecular level, like what's the underlying mechanism that's happening right when we spray with Preaxor. But I think that the downstream effects are pretty well established across different species and in wheat. So. Yep.
Thank you. Next up, I'd like to present our next student speaker, Andrew Felton. He's a PhD student studying ecology in the Smith's lab. Please welcome him.
All right, thank you all for coming. I'd like to thank Ray for inviting me to give this presentation. So I'm going to go from the mitochondria to the ecosystem level. I guess that's what I was recruited for this talk. So I'm going to try to give a bigger, larger scale perspective on different components of drought. And specifically, I'm going to be talking about interactions between changes in intra-annual variability in precipitation, so within growing seasons, and interannual variability in precipitation, particularly within the context of drought. So this is all pretty new stuff, i.e. my advisor does not know I'm here, or I'm talking about this, or even about this project. So I think this is a good platform to get some insights and constructive criticism. So on that, I will begin. So with climate change, rainfall regimes are predicted to change at multiple time scales, so among and within years. And within years or among years, we're expecting greater frequencies of wet and dry years and increased frequency or intensity of climate extremes, such as drought. Yet at the same time within years, there's predictions of increased variability in precipitation event size and distribution. So specifically, larger yet fewer events are one of the key long-standing predictions for changes in rainfall regimes as we intensify the hydrologic cycle and increase the water and holding capacity of the atmosphere. So for this talk, I'd like to focus on mostly within years and the consequences for changes in within year precipitation, the consequences for that, to ecosystem responses to changes in interannual variability in precipitation. So this is a study by Alan Knapp, who many of you I'm sure know. This is kind of a groundbreaking study, and it's almost 15 years old now. But he manipulated within a MESIC system the event size and distribution of rainfall. So he imposed larger yet fewer events. And the general takeaway is that the effect of this manipulation, independent of changes in total rainfall amount, decreased multiple carbon cycling processes. So net photosynthesis, above ground net primary production, so CO2 flux, and a mechanistic link with this is increased ecosystem water stress. And so when you look at the effect of the treatments on soil moisture, there's a stronger link with above ground net primary production to the variability in soil moisture than to just mean soil moisture content. So the general takeaway from this is that increased variability in within year precipitation can actually reduce carbon cycling and productivity within this mesic ecosystem. And so changes in productivity independent of total precipitation amounts could suggest that there could be heightened sensitivity to water limitation, such as drought, in the future. And so that leads to my key research question. How will changes to intra-annual variability in precipitation interact with changes at the interannual level? And so I took what I believe to be a somewhat unique approach to trying to answer or get at this question. So I did a manipulative experiment, and then I compared the results to that to the long-term record. And so I did my manipulation at the Konza Prairie Biological Station, which is very beautiful grassland. I now have appreciation for how beautiful grasslands are after working here. So this is a great place to work because you can do manipulations, but there's also a long-term record of productivity and precipitation. So you can put your manipulation, the results of that, in the context of longer-term ambient trends. And so my experimental approach was to exclude all ambient growing season, so May through August rainfall. And then I went out into the field and I performed weekly water additions to reach specific target precipitation amounts, which I'll show you in a second. So I literally spent my summers watering grass. Hopefully it was worth it. So to essentially eliminate the variability of growing season precipitation, I kept the timing between rainfall events essentially constant, either 6 to 7 days, or the event size too, which was just the monthly mean divided by 4. So I try to keep these two components, event size and distribution, virtually constant. And I deployed that manipulation across an entire rainfall gradient. So instead of just having 2 or 3 levels with multiple replicates, I exchanged replicates for number of treatment levels. So I performed this in 2015 and in 2016 in separate neighboring sites. So in 2015, I had 9 levels, and in 2016, I had 11 levels. And these levels range from the first percentile, so the driest conditions on record, to the 99th percentile, to the wettest conditions on record. And each of these levels had 4 replicates. So just to give you a sense of how I carried this out, I built these rainfall manipulation shelters to fully exclude, and then I added water under them. So I deployed this manipulation on a fairly large scale. So just to give you a sense of the differences between my treatments and the long-term record, so in the open circles is the variability from my experiments for both years, in the open circles, that's the ambient record. And so you can see in that first graph on the left, the standard deviation of event size, or just the variability in event size, was reduced by quite a bit overall, about a 92% reduction I was able to do with my manipulation. And then with that, mean size is pretty much linearly correlated with the amount of growing season precipitation I added, but there's more of a saturating effect in the long-term record. So I'm just trying to give you a sense of the change in variability that my experiment output. And so for this project, I'm focusing on rain use efficiency. So it's pretty much just the ratio of net primary production, so what was produced that year, net carbon inputs, to growing season rainfall. So how efficient was ecosystem productivity to total growing season amounts? And just to be clear, to get kind of a robust assessment of this, I combined years to get, so I just put the average of these points to yield 1 point. And so I did this for all points that were connected to each other, and then I calculated the mean of these guys too for productivity and precipitation to try to get a robust estimate around the mean, or the 50th percentile. And the general takeaway when you look at rain use efficiency with my manipulation, where you reduce variability in rainfall event size distribution, is that there's pretty minimal effects at the wet end of the spectrum. But as conditions get increasingly dry and transition into severe drought, the nonlinear model, which both of these are best fit for, predicts a 63% increase in rain use efficiency when you completely remove intra-annual rainfall variability. So removal of rainfall variability had minimal effects during wet years, yet increasingly large effects during the driest years. And so because we saw these effects increase as water limitation increased, I focused on ambient rainfall regimes during years of 500 millimeters and under. And within this, I split it up further into the 7 driest years and all other years. So any years under 300 millimeters and then 300 to 500 millimeters. And I looked at attributes of these years in rainfall variability to see what factors related best to precipitation use efficiency. And so initially, I considered almost 20 to 30 different things. I spent a lot of time looking through the data, but these were kind of the key ones I landed on considering. So just the mean event size, the average number of consecutive dry days, the standard deviation of event size, maximum event size, and then April precipitation to provide some inference on antecedent rainfall, since I'm focusing on May to August. And across all these years, when you first look at it, it's really maximum event size that the model selected as the sole predictor of precipitation use efficiency. And so maximum event size was very highly correlated with the standard deviation and rainfall variability. You could say they're collinear, but at the same time, I would suggest that maximum rainfall events, so these large rainfall anomalies, are generally predictive of increased variability in rainfall event size. So in the years when you have these anomalies, you're going to have more variable rainfall event sizes in potentially just in general rainfall regimes. And so I was interested to see if this would hold up if I just looked at the 7 driest years on record. And it didn't. The model selected pretty much everything, so there wasn't a clear signal in what drove precipitation use efficiency in the driest years. And although there was a generally significant negative effect of maximum event size on rain use efficiency, there were clear outliers to this. And so I looked at this. This year particularly had the lowest variability, the lowest maximum event size, and also had pretty low rain use efficiency. It also, in general, had very low rainfall amounts. And in that year, it had among the lowest April precipitation inputs in the historical record. And so this suggests that while more variable rainfall events may reduce precipitation use efficiency, when you get into the driest years, there may be added complexity and contingency in this, such as antecedent soil moisture or other things. And so relating this to previous findings, so that graph I showed you at the beginning, so that study found that the largest effects of increased intra-annual rainfall variability, the largest negative effects, occurred during the driest years. So that adds some support to what we're seeing here. But in a modeling study, Shi et al., they found that during severe drought, this ecosystem was more sensitive to reductions in event size. And they also suggested that during the years of the most severe drought, that large rainfall events may actually increase rain use efficiency of the ecosystem. So there's some conflicting insights here, but I just would suggest that a potential future area of good research and interesting research would be to look at characteristics of rainfall regimes during years of severe drought and to see how that affects the sensitivity of ecosystems to broader scale interannual drought. And this may be particularly true when we go across different ecosystems. So while increased variability, larger and fewer event sizes reduced productivity in the music ecosystem where I worked in, that similar type of manipulation has actually increased productivity in semi-arid grassland that we have out here over at Pawnee. So ecosystems are going to respond differentially to changes in intra-annual variability, and that could have potential consequences for their responses to severe droughts if intra-annual variability and inter-annual variability interact with each other, which I would suspect that they will. So some potential implications I'm thinking about in this project is that the positive effects from nearly completely removing rainfall variability on the rain use efficiency of ecosystem productivity became increasingly large as conditions transitioned to severe drought in the driest years. This could potentially suggest that if you have higher intra-annual variability in this ecosystem in the future and more severe droughts as well, this could reduce the rain use efficiency of this ecosystem, reduce its resistance to severe drought. So there is some evidence from the long-term record that larger rainfall anomalies drive increased variability in precipitation and reduce precipitation use efficiency. However, intuitively, I think, and from that one outlier, that when you get into the driest years, there may be added contingency and sensitivity of the system to changes in different components of rainfall variability, such as April precipitation. So it'd be interesting to get your insight on that. And that's all I have, so thank you. Yeah. So I did it over 2 years, so probably not. And I actually deployed the same manipulation this year in semi-arid grassland. So I've kind of moved on from this system and I'm looking at potential implications within semi-arid grassland out at Pawnee. So.
Did you, maybe I missed
Yeah.
this, did you have measurements of like EPD or soil moisture?
Yeah, I had measurements of soil moisture, not BPD.
Okay, and that wasn't an important correlate or descriptor of NPV in the end?
Oh, it totally is. I just haven't focused on it here. But yeah, it's definitely.
Mechanistically, that seems to me to be like those two parameters might be your strongest drivers.
Yeah, and I would argue that soil moisture is the strongest driver. Because, yeah, I have generally looked at it and did a model selection. Yeah, it's all about soil moisture, is what I've concluded, or at least what the stats would suggest. Yeah.
And the lower precipitation use efficiency, when you under extreme rainfall events, is that low because water was not taken up then? Correct?
Yeah. So
where's that water going?
So there's different theories and ideas. You could have surface runoff, percolation down to deeper layers, and it just gets lost. The general idea is that when we get these larger rainfall events and fewer of them, they percolate down to deeper soil layers, and they're not getting fully utilized in the top 50 centimeters, where about 70% of the rooting biomass is in this ecosystem. So that's kind of the general idea of it. Yeah.
In your relationship between maximum event size and variability, is it that the one event size driving that variability? Like if you remove the maximum event size, would the precipitation variability be different between years? I'm just curious, is it that one event that's really driving that?
I would suggest probably, yeah, but in these years that have maximum event sizes that large, they generally have a couple too. So yeah, it would be interesting to look at if you just remove the maximum and do like the next one, something like that. But yeah, I mean they're collinear essentially, so they're predictive of one another. So I would guess that yeah, that's generally what's driving the variability. Yeah.
Did you look at when the max events are happening? So if you have a year that has a very large maximum event, but it falls in May, is that more important for rain use efficiency than if it falls in September?
Yeah, so I've just started to look at precipitation regimes during those years. So I'm kind of like on that path. I kind of suspect that they're probably more important for productivity in like June and July maybe, versus if they fell in August or something, they may not really be used that much towards productivity. But yeah, it's something I'm starting to look at. Okay, thank you.
Our third and final student speaker is Taylor Pearson. Taylor is a master's student in the Department of Bioagricultural Sciences and Pest Management. She's studying sorghum in the Corey John Lab. Taylor is also one of the co-organizers of this event. And she's going to mix it up a little bit today and talk about an overseas trip that she did as part of the National Needs Fellowship. Is that good on the mic?
So like Heather mentioned, I am one of the National Needs Fellows, and I'll be talking about my international experience as a requirement for this program. As a National Needs Fellow, we have several requirements to graduate as a fellow. There are 3 of us. One of us has already graduated, Craig Beal, who's actually watching the live stream right now. And all of us were required to go abroad. And Craig, he went to CIMMYT, which is the International Maize and Wheat Improvement Center in Mexico. He went there in spring of 2015. And Gray will go, he's planning to go to the International Rice Research Institute next semester. You'll see his poster is up here too, so you can see it during the poster session. And myself, earlier this year, I went to ICRSAT, the headquarters, which are situated in India, which I'll talk about in a little bit. And essentially, this program is a fully funded, minimum 3-month experience at some international research center. So loosely, most of our options for this international internship are within the CGIAR network. And CGIAR is the consultative group on international agricultural research, which consists of 15 nonprofit research centers with the overarching goal to innovate to help the poorest people in developing countries. And there's a lot of ways that these problems can be addressed, but I've listed those that are most relevant to the topics of the symposium, climate, water, crops, and most importantly, policy. So with this, I went to one of these centers, which I mentioned is the headquarters of ICRSAT, the International Crops Research Institute for the Semi-Arid Tropics. So you see this is a map of India, and where the red circle is, that is where the center is in Patancheru, India. And this is really an ideal location for an agricultural research center because there's a lot of environmental variation. We've already mentioned a lot of these things, but vapor pressure deficit varies throughout the year. Precipitation, you have your rainy season, you have your dry season. And there's 3 full growing seasons per year, so you can cycle through a lot of projects, a lot of germplasm. So a lot of potential at this research center. Another benefit is being able to hire a lot of people for a low price point compared to other countries. So you can really maximize the amount of research you can do in a single year at one time. And lastly, there's a lot of partners and projects and collaborations with these international centers. 2 of the largest patrons for the projects that I was working with were under the Bill and Melinda Gates Foundation and USAID, which I'll mention 2 projects under the USAID Feed the Future initiative. So I was at ICRSAT for 3 months and 10 days, and I was in the plant physiology lab under the advisorship of Dr. Vincent Vedez. He's pictured there on the far left, second guy on the left, and he focuses on dryland cropping systems. And Dr. Vedez studies many crops, all that are listed here, both cereals and legumes. I had the opportunity to work with most of these as well. And you notice highlighted in bold are the crops that are mandate crops for ICRSAT. And Dr. Vedez is really great in recruiting people from all over the world to come visit in his lab and collaborate. And this was definitely the case when I went there earlier this year. I met folks from all different countries of the world, and I think in our lab we spoke 11 or 12 different languages between everyone. So a very dynamic group. And now I'll get into just a few projects that I spent the most time working on during my internship there. I think the coolest and most novel piece of technology I worked with was what we call the LESI scan. Here's an aerial image of high throughput technology. And this is really unique in that it's high throughput at field density. There's a lot of high throughput technologies that are even in a controlled environment or they're looking at a single plant. So this is really great in that you can look at in field conditions, although they're potted, they're planted at field density. And I'll show a short clip in a bit and describe what these parts are, but essentially they're scanners. They send out a laser based on the reflectance. You can kind of get an idea of how big the canopy of the plant is or the planting density area. And this is collected continuously and simultaneously with environmental data. And this environmental data is wind, precipitation, humidity, photosynthetically active radiation, all of these things. So you can determine how the plant's responding to both diurnal variation, during the peak sun hours or in the evening or the morning, as well as seasonal variation, early season, late season. And this is particularly important when we're studying drought-related parameters, namely heat, vapor pressure deficit, and of course, water stress. Another key part that was recently added, I think in the past 1.5 or 2 years, was the ability to untease evapotranspiration. So you know how much is evaporating from the plants or from the pots, and how much water is lost throughout the plant itself. And they're able to do this by having load cells under each pot or tray, which is essentially a scale. And these measurements are taken every half second. So you're getting this continuous data. And I'll have to go through this a little bit quick, but I'll have my poster up if you have any more questions about this. And essentially it's on these scanners are on a gurney that's moving on wheels, and it could scan 14,000 plots per hour, moving at 3 meters per minute. So here's a short clip. It might be a little shaky. I was a little drought stressed taking this video. But you see these white boxes. This is where the laser's emitted, and it's getting that canopy reflectance data. They can be adjusted to whatever height based on how big the plants are, how low they are. You can see we're growing legumes and cereals, mung bean, pigeon pea. Yeah, sorry for the shakiness. And you see these little metal bars here. These are essentially a barcode. So as the LASI scan is going across all of the platform, it knows what sector it's scanning. So really cool technology. I was I'm very grateful to be able to work with that. So this technology is all well and good, but we need to make sure that it works. So essentially, in 2 6-week periods, we had 2 plantings, and we had to manually validate what the machine was giving us to what actually the leaf area was. So you can see us harvesting up there in the top left, just the process. We get the plants, bag them up. In the bottom left, we are working with chickpea, which is probably the most challenging plants to look at leaf area. We actually had to put the leaves on transparencies and then scan them because they're so small. And of course, our team in the bottom right. But we essentially wanted to validate what the machine was giving us. And yeah, we compared those values to what we manually determined. And as you can imagine, there's a ton of data to process when you're doing this, getting weights every half second and reflectance data. So this is still in progress. I don't have any results for you now. Another project that I worked quite a bit with was under the project lead, Lee Jalan Balcha, who's from Ethiopia, Addis Ababa University, but now he's at UC Davis collaborating. And this is under the USAID Feed the Future initiative, and they're looking at chickpea. It's a pretty important crop in a lot of semi-arid areas. And we're seeing whether or not, we're just characterizing chickpea branching. So we look at primary branching, secondary, and then tertiary. So a lot of characterizing branches. You close your eyes and you see chickpeas. A lot of time there. But he's really trying to ultimately integrate wild-type drought resilience traits, perhaps chickpea branching morphology, into cultivated lines. And this was the first year of this evaluation, so still a lot to be analyzed for that. Another trait of interest under the USAID Feed the Future initiative was stay green in chickpea. This is Eddie Marquez's project. He's also, he's out of the University of Vermont. And as John Mullet mentioned earlier, stay green has been very favorable for selecting for drought-adapted sorghum varieties. So they're looking at the same thing for chickpea. And after evaluating a lot of water use efficiency things, these are lysimeters, which can measure that kind of trait. Eddie found that the stay green in chickpea, at least, for what they evaluated in these lines, was purely cosmetic. It had no effect on drought resilience. And lastly, I did a little bit of shubelomics work with Regina van der Valk from Belgium. And she was looking at a subset of parents from a population, evaluating the root traits of different sorghum genotypes and seeing if they were different. So we looked at root architecture, which is what shubelomics is, and she characterized 32 different genotypes, both at the 6-leaf stage early on and at flowering as the plants are here. And we looked at a slew of root and shoot traits. And as you can see on the image on the left, it's kind of like a protractor. We measured crown root angle, which is a pretty good indicator on how the plant's growing under water limited conditions. So usually if you have a steeper angle, the roots are trying to reach the deeper profile for stored water, which might be valuable in an area where you get, a monsoon season and then a dry season. And just some take-home points. Regina, she just finished with her master's this summer. So some take-home points from her research. She found that the nodal root angle is associated with increased yield in a semi-arid environment. She also found that it's genetically independent of other traits and highly heritable. And when we're talking about the different kinds of roots, it's not as important, at least for these genotypes, to distinguish between nodal roots, those that come from the base of the stem, crown roots, those which come from the base of the stem, and nodal roots, which come from the first node on the stem. So I worked a lot. Pretty much most of my time was involved in these research projects. It was a very dynamic environment, like I mentioned. But outside the lab, in addition to dreaming about Reuben sandwiches in the land of rice and vegetables, which happened a lot, I was able to attend the Interdrought 5 conference, which took place in Hyderabad, which is about an hour south of the ICRSAT campus where I was. And I was able to share my research, listen to several talks, some of which were mentioned in the earlier presentations, and of course met a lot of great people from all around the world. I was also able to do a little bit of extension work. Here I'm at the LeasyScan platform, and I'm talking with a group of middle schoolers about transpiration efficiency and trying to put that in terms that a 12-year-old can understand. Harder than you think. But it was really great to talk to kids about what I'm studying, because it really brings it back to the big picture of like, why am I doing this research? And lastly, I think the biggest take home was the people I met. You're working with these people, living with them for months on end. And I think you gain something that you can't get from watching a documentary or reading any kind of, published paper. I remember, like Lee Jalam, for example, we spent time making sure rats didn't eat our chickpeas, and he was telling me all about the problems facing Ethiopia and the sustenance farmers. So you just get a better understanding of what's happening in the world, and especially the problems facing agriculture. So with that, I really want to appreciate and thank the National Leads Fellowship Program for funding our international experience, and I hope others have the opportunity as well. Thanks. Questions?
So you had mentioned earlier that there are several mandate crops that you have primarily focused on. Can you explain what characterizes them as mandate crops?
Yeah, good question. So mandate crops are pretty much the responsibility of a certain center based on the regional need. So those mandate crops are those that are grown by sustainable or sustenance farmers, which that's their primary food consumption or the only means that they can make a living. So it's the responsibility on these centers to improve these cultivars to ultimately improve the livelihood of those poorest farmers, stakeholder farmers. Okay. Thanks.
Thank you so much. We're going to break for an hour for lunch. We're going to come back at 12:40 and introduce our next keynote speakers then. Thank you.